Thank you all for coming. This is amazing. This is the third Foodscape session we've done at SRF. The first one was 2019. The last one was 2022. So I think we're on a 3-year cycle. So 2028, make sure to mark your calendars for that. And every time we've done a Foodscape session at SRF, I was like, this is the highlight of my SRF career. It can never get any better than this. I should just quit now because it's been amazing and everyone's been so supportive of it. And every time I've been wrong. And looking at the lineup of the talks today, I know I'm wrong again because I'm like a kid on Christmas Day waiting to hear all these talks. These talks are going to be amazing. And you're going to love this, I think. I'm going to love this. So I have like 12 minutes to go through the session protocol and then try to introduce the Foodscape concept adequately to lay a foundation for the rest of the session. So naturally, we're going to start with some Qigong breathing. So everybody put your hands on your belly, right? Take a deep breath. Fill up your Dantian. Breathe out. And if you make your hands like claws, right? And you just start tapping on the top of your head, like get in there real good. Get into that occipital ridge. Make it hurt just a little bit, you know? Forehead. Get everything moving. Yeah. See, this is great. Everyone's going to be ready. Everyone's ready. We're all like 10 times smarter than we were 5 minutes ago. Okay, so session protocol. We have 23 minutes for our talks, so we're trying to keep them around 20 minutes, so we have time for 1 or 2 questions, but we're not probably going to have time for a lot of questions because we have a very full day, 14 talks. I have these little, they look like when you're at an auction or something, auction battle. I'll tell you 5 minutes, 1 minute, 0 minutes. I'll start playing Justin Bieber when you run over time, your outro music. And we'll save longer questions for tomorrow tonight or during break, so it's going to be great. So the Foodscape, where do you start talking about this? I've always been really fascinated between the relationship between a novel and a scientific model. And at one level, you might think, why there's no relationship between a novel and a novelist can just make up what they're writing about, and a scientist has to be true to the facts of the system that they're studying. But I think if you look a little closer, that's a superficial distinction, because all the really great novels, even the most fantastical sci-fi novels, have to be true to the human experience at some level, right? That's why they resonate with millions of people. That's why they're kind of universal in some ways. And the really good scientific models have to engage with the human imagination. They have to kind of tell a story in a way that we haven't previously told the story, so that we can make connections and we can generate hypotheses that we would not have otherwise generated. And I think this is definitely true with the Foodscape. The author Milan Kundera, whose work I've never read, admittedly, but was a novelist and was up for the Nobel Prize in Literature, has this quote I wanted to read about a good novel. He said, "Every true novelist listens to the supra-personal wisdom which emerges from a body of people, which explains why great novels are always a little more intelligent than their authors are." And he said, "A novelist who is more intelligent than his book should go into another line of work." And I think that's true with a good scientific model. And I think it's true with the Foodscape and also the process of writing the Foodscape paper, which many of the authors of that paper are in this room today. In a way, the Foodscape story and the Foodscape hypothesis and model, if you want to call it that, is smarter than all of us individually. It's certainly smarter than me individually because it generates connections and hypotheses that we did not have a priori. Things emerge from it that we did not expect when we built the story. And so in that way, it's generative, like a good story is generative, but not like the creepy AI kind of generative. It's the kind of generative that emerges from a body of human beings who are focused on the same story, and they all share collective experience, but they share it together with the same focus. And so then in that body of people, something can emerge that's more useful than any of the individuals have. So what is this? What is the Foodscape? What are we talking about? The Foodscape brings together 4 elements of river ecosystems. And it tries to see them in time and space. So those 4 elements are, first, the production of food across a riverscape, across the watershed, into the marine environment. The second is the accessibility of that food to animals who are moving across that environment. And the third is the relationship between the physiology of the animal and the environment where they are. And those 3 things interact collectively to create what we call growth potential, the capacity for an animal to grow in the landscape. And then the fourth element of the foodscape is trying to see that growth potential across space and time through the eyes of a mobile consumer who is moving across these landscapes, which is a really like human thing to do. We always try to see the world through the eyes of something that we're not. You know, each of these elements of the foodscape is like a universe. You could dive infinitely into any one of them and get lost. And when you look at the interactions between them, it's like a metaverse. But there's a method to the madness. So let's just start briefly thinking about food production for a second. Food, of course, can be thought of in many different ways. We can think of it as any nutritious thing that an animal eats and assimilates. And the assimilate part is important because fish in particular eat a lot of things they don't assimilate. They eat sticks and algae and very small rocks, to quote Monty Python. And in our models, especially our bioenergetics models, we often turn food into calories, right? We turn food into calories or joules. But we know that that's not adequate. We don't think about our own food that way, right? When I go to eat food, I'm not just like, oh, how many calories are in this? Food is all the micro and macronutrients that are essential for life. A good example of this is my advisor, Mary Power, has this love affair with a very small diatom called epithemia. Diatoms are these single-celled algae encased in silica. And if you look at a microscope, you know, they look like cosmos. They look like the universe. Like they look like some star constellation millions of miles away. My wife says God is boring. He just finds a good pattern and repeats it everywhere. But epithemia is interesting because it synthesizes long-chain polyunsaturated fatty acids. These are when you go to the health food store and you want to get fish oil and you see the DEA or the EPA, that's what these are. And PUFAs is what they affectionately call them. And PUFAs are essential to the brain development of every invertebrate animal on Earth. If you have a brain, your brain functions because of polyunsaturated fatty acids. I recently saw a paper that correlated the PUFA content in muscle tissue of brown trout to brain volume and behavior. And these have also been correlated to survival of juvenile emigrants, right? And this is just an example of the infinite different ways you can think about how food moves through an ecosystem and how consumers use it. So if there are other things that are rich in PUFAs, right? So the gammarid amphipods in the estuary are super rich in PUFAs. And salmon eggs themselves are super rich in PUFAs. So if we close your eyes and try to visualize a watershed and you think, where are these things generated from? How do they move, right? Are they synthesized in the same place that they're consumed, or do they transported some way? What facilitates that transport? How are the life histories of animals that ultimately eat these things adapted so that the animals are in the right place at the right time to take advantage of that? And most importantly, how have we altered watersheds so that we are altering the relationship between the trophic pathway and the consumer? And this is what I think is novel about thinking about foodscapes and food web restoration as a driver of salmon recovery. Is so much of what we thought about for salmon recovery has been emphasizing the physical habitat, which is very important. We know habitat structure, in-stream flow, water temperature, all those things matter. But this element of the way the animals relate to each other and the way they relate to the food they eat is fundamental to their population abundance, to their, as we're going to see in talks today, to their resilience, to their recovery. And it's not something we've thought enough about in terms of how we work on restoration. Wheatley and Baxter, in their 2010 paper, which we really love, they organized riverine food into 4 groups, right? So there's food that's produced in situ, right? That is, so think about a standing crop of invertebrates. There's food that's transported from upstream to downstream, so think about invertebrate drift that we all hopefully think about. There's food that's transported from downstream to upstream. Did I say that last time? I meant upstream to downstream last time. That's drift, right? Downstream to upstream would be like a marine subsidy, something coming up in the body of a lamprey or a sturgeon or a salmon. And then there's food that moves laterally from the terrestrial environment into the aquatic environment, infall, drift, when you inundate a floodplain and you entrain food that was growing in the terrestrial environment. And each of these broad groups kind of hosts an infinite number of different individual trophic pathways. And it can be dizzying and probably too much, and we probably shouldn't even try to map every single one of them. But if you start thinking about them, and you start thinking about the watershed you work in, it's very possible that there were keystone trophic pathways, really important trophic pathways that fueled production of fish in your landscape. And those may have been altered by how we've, when you build a levee, you're not just altering rearing habitat, you're altering the transport and the flow of food between the terrestrial environment and the aquatic environment. When you build a dam, you're not just altering in-stream flow and sediment, you're altering the movement of food and the movement of the constituents of food, but you're also altering the movement of animals to access that food. And the whole coevolution between the animal and the foodscape that is moving across. So this is foodscape thinking, and when you do this, you're doing foodscape thinking. And the same kind of thinking applies to the other attributes of the foodscape, right? So food accessibility relates to everything that affects the ability of a consumer to access the food that's produced. This might be turbidity, right? That's an easy one, right? It might be water temperature, that's either preventing or allowing animals to get into different rearing environments. Dissolved oxygen drives that. But it also might be biological interactions, right? A large predator or a piscivore might create a geometry of fear that prevents or facilitates animals from accessing food. And then the physiology of the animal, which is kind of the traditional scope of growth we think of for bioenergetics, is the relationship between things like water temperature and the internal physical processes of a consumer, which allow it to grow, facilitate its growth or not. So once you've mapped this out in your watershed and you think about these, then you have to do the work of thinking about how does an individual consumer or a population of consumers move across the landscape to take advantage of really what is kind of becomes a heat map of growth potential, where different parts of the watershed are going off at different times of the year. I've often, it's not very, what's the right word? It's not very honorable to salmon maybe, but I've often compared salmon to a slime mold, right? Like a slime mold will spread itself out across a landscape and try every different little way to get to the different food. And once it finds pathways that work, it'll put energy into the cells of that way. And maybe an individual little salmon isn't the smartest thing in the world, but a population of salmon trying all these different trophic pathways and trying them over time so that their life histories become co-evolved to, so their emigration timing, to their growth rates become co-evolved to the heat map of food resources across their landscape, becomes a functional driver of resilience and abundance of that population. So, since, even before we worked on the Foodscape paper, and like scientists, we come to slap a name on something that already kind of exists in the body of people. There were people that were working on this for many, many years. And everybody I cold called to talk about Foodscape was like, oh my gosh, yeah, this is what I'm thinking about. The paper kind of wrote itself in that way because people were so excited about it and had already been thinking about it. But there's been a renaissance of work, I think, in this field in the last maybe 10 years. And what is great about your talk today is you're going to hear, we call this Foodscapes in Action, because you're going to hear about folks doing fundamental research on this, but also doing applied restoration on this work, who are applying these ideas to recovery processes and recovery actions. So, yeah, I couldn't be more excited to hear these talks. I'm just going to sit down like everybody else and soak it all up. But I'll have to make sure I keep the timer going, and also not get distracted by this incredibly beautiful view. We're actually going to start, rather than on the production of food side of this, we're going to start with the animals. I'm going to introduce our first speaker really quick. I have to pull up my speaker intros.
Okay, we got a little situation.
Jumping around. Well, that's good. While you're fixing that, I'll introduce our first speaker. So our first speaker is Stephanie Carlson. And Stephanie's a fish ecologist at UC Berkeley. Her work focuses on understanding the dynamics of Pacific salmon and trout, including the factors that shape populations and influence their persistence. And a long-running research theme of Stephanie's work is the study of habitat mosaics, connectivity, life history diversity, and as factors that support abundant and resilient salmon populations. And it's a theme she's going to address in her talk today. And Stephanie was one of my Ph.D. advisors, one of my favorite scientists and favorite people, so I'm just like really, really thrilled to hear these talks. So I'll turn it over, Stephanie, to you.
All right.
It's amazing to see this room so full. I'm so excited to speak to you all. Can you hear me all right in the back? Okay. Yay!
Good morning, Stephanie.
Well, for those of you who are just learning about the Foodscape concept for the first time, I'd also encourage you to check out the supplement to that paper where one of our heroes, Mary Power, wrote a song describing the Foodscape concept. And it's included there in the supplement. Somewhere over the Foodscape, hot spots boom. Somewhere over the Foodscape, I'll find my dining room. Gabe, Ryan, and I are all singing backup on it, so ignore that part. But anyway, it's amazing to be here. chance to share this work. We've been chewing on these ideas for a number of years. It's incredible to work with this group of people. All of them have shaped the ideas. Even more incredible is that every single one of them is here at this conference. It's very special to spend time with them. And I'll just say that collectively we've been on the case of the missing life histories. This is a little awkward, but I'm going to start by stepping back and pointing out this essay that was published about a year ago, in which Bilby et al. Emphasized that following the listing of several species of salmon and trout in the 1990s, that there was just this rapid development of the restoration sort of industry and extensive amount of habitat restoration that's happened since that time. And they point out that despite billions of dollars, literally billions of dollars that have gone into habitat restoration, that most of those listed stocks are not improving. And they ask the simple question why that might be. And they present 5 ideas, and this list is by no means exhaustive, but a few of the things that they point out, for example, are that the scale of habitat loss and degradation just far exceeds the amount of habitat that's been restored. We simply haven't done enough of it. They also point out that monitoring has been inadequate, often too short of a time scale to really detect population level impacts. To that, I'd add that we've done far too little monitoring. There have been missed opportunities to study these restoration interventions, to learn what's working, what's not working, and to improve upon it. But the main idea that I'm going to focus on today is related to their point number 2, that we're not doing the right things in the right places at the right times. In other words, we're missing some things. And so I want to talk about our ideas about one thing that might be missing. And I'll just start by emphasizing that we made this observation that many basins seem to lack the natal stream capacity to produce the historic adult numbers. It tells us that we're missing something. And if I zoom in and focus on the Eel River of California, Northern California, for example, the system historically supported runs of coho of up to 150,000 adult coho and up to 200,000 adult steelhead. And yet the perennial natal stream habitat that yearling smolts would rely on is quite limited in the eel, and likely was historically. And if we sort of zoom in on the South Fork of the Eel River, where we think approximately a third of these steelhead would have returned, maybe 60 to 70,000 fish, we can just sort of work through the math and try and figure out what might be missing. So in the South Fork of the Eel River, there's almost 700 miles of stream channel, but only about 150 miles of this perennial cool habitat that would support yearling smolts. If we zoom in on those 150 miles of habitat and we seed them and we apply really high early life survival to those fish, we might generate estimates of perhaps 1,500 smolts being produced per mile. If we then multiply that by the number of stream miles, we might think that this system generated historically maybe 225,000 smolts. If we then apply an ocean survival rate of 5%, that would lead to 11,000 adults returning. Maybe we apply an even more optimistic ocean survival rate of 10%. That gets us to 22,000 adults returning. But we think that 60 or 70,000 adults return to the system. So our question is, how did they do it? What are we missing? And our main hypothesis is that a lot of fish left those streams early in life, spread out on the landscape, and used an array of productive stopover habitats elsewhere in the system to rear and grow before migrating to the ocean. And that these habitats that supported these non-natal, so-called non-natal life histories, have been disproportionately lost and degraded. And consequently, our main hypothesis is that recovering salmon is going to require recovering those productive stopover habitats and access to it to recover these missing life histories. This is basically my talk in a nutshell, and now I'm going to unpack it for you. But I'll start by just sort of stepping back and pointing out the obvious, which is that streams have limits. We know that streams have carrying capacity, limitations. For example, there's dozens of studies that show patterns like this one. When there's more fish, they grow more slowly. When there's more fish, they experience lower survival. And consequently, there are limits on the number of smolts that can be produced in any given natal stream environment. If we sort of zoom in and think about any one particular system, what we know is that if you run a smolt trap in any one of these systems, we're targeting these 1+ fish shown here. Our monitoring programs are focused on understanding the number of 1+ smolts that are produced in these systems, and yet we know that when you run these traps, you catch a ton of these little guys, leaving 2. And in fact, in this particular system, the South Fork Sprout Creek, what we can see is that there's an order of magnitude more fry produced in this system than these targets of our monitoring program. And so while we're focusing our monitoring here and focusing our restoration actions by putting wood in streams to create pool habitat that supports these fish over the summer and over the winter, what we're kind of recognizing is that there's many more of these fish that leave that habitat, and we know very little about where they go and their success. And I also want to point out here that remembering that streams have a carrying capacity. If these little guys didn't leave, just imagine how intense that density-dependent survival would be, right? The fact that those fish leave improves the environment for the fish that stay through alleviation of density dependence. We also know from there was a lot of people who were kind of obsessed with these fry, kind of like me, in the 1970s. There was some work done in Oregon in the Rogue River system by Fred Everest. I really like this study. He marked over 30,000 of these fry migrants from a tributary of the Rogue River. And then through his recapture efforts, he recaptured those fish in different locations. And what he found was that some of those fish moved downstream and took up residence in the main stem of the Rogue River, just downstream of the tributary in which they were marked. Other fish moved downstream to the Rogue and then moved up other tributaries, showing some degree of habitat choice. And then a fraction of these fish were recaptured a year later as yearling smolts. So all of this suggests that these fish aren't simply surplus production lost to the population, but in fact, when they encounter suitable habitat, they do well, they survive, they contribute to the smolt production. And he hypothesized that those fry that left the system were the offspring of late spawning adults, and that the offspring emerged into an environment that was already fully seeded, and consequently they departed that environment for greener pastures elsewhere. So density seems to be one of the cues driving that downstream movement. We also know that flow and temperature can trigger these downstream pulses. This is some work by my predecessor at Berkeley, Don Ehrman, working in the Kiln Meadow tributary, which is a tributary of Sagehen Creek up near Truckee. And with his student George Leidy, they were basically studying these pulses of rainbow trout leaving the Kiln Meadow tributary following the snowmelt. So this is a snowmelt-dominated system. Rainbow trout are breeding in the spring, and then their offspring are moving out with the snowmelt and as water levels decrease. And in many years, the system goes totally dry in the summer. So this is also a reminder to us that some places are important as breeding habitat only. But for those fish to be successful, they have to find suitable habitat elsewhere. They leave early and seek out suitable habitat elsewhere. So from this part of the talk, I want you to remember that fish are moving early in life. They're redistributing themselves across the landscape, and in moving out of these natal streams, they're actually alleviating density dependence in that natal environment in ways that benefit the natal rearers. Linking back to the Foodscape ideas that Gabe introduced, I also want to highlight that as those individuals distribute themselves across the landscape and encounter different parts of the foodscape, that they're also going to be encountering, different temperatures, different food webs, as Gabe mentioned, different predators and competitors, and this is going to influence their survival and their growth. So in particular, thinking about the food web production, we can sort of think about the fact that there's asynchrony in those production patterns and different habitats, and as different individuals move through the landscape and interact with different parts of the foodscape, they're going to grow in different ways. And they're going to move through their life cycle in different ways. And in particular, they're likely to reach the ocean at different sizes and different timings. And these are two traits that we know have a strong influence on their early life survival. More generally, from kind of contemporary salmon conservation science, we also know that when individuals move through their life cycle in different ways, that this has benefits for the abundance and stability of the adult population complex. So the classic work on this topic comes from the Bristol Bay system. These are data on sockeye salmon from the Bristol Bay region, showing 3 different components of this fishery for sockeye salmon. And in this particular paper, the authors argue that it's important to maintain this full suite of populations that differs in their life histories and differs in their dynamics as a way to buffer against environmental variability and change. And in particular, they point out this 1977 PDO phase shift, where ocean conditions changed considerably. And what you can see is that this igigig stock that was only a minor contributor up until that point suddenly kind of turned on and became a major contributor after that ocean regime shift. And so they emphasize that maintaining that full complement of populations buffers this population complex to that environmental variability and change. It's also important to recognize that in every year, individuals from each of these components contributes to the adult returns. So abundance is also influenced by this life history diversity. Life history diversity contributes to abundant and stable population complexes. Sorry, it's a little awkward. So just as a reminder here, remembering that this life history diversity within and among populations is increasing both the abundance and the stability. We want to be managing for it, trying to restore it where it has been lost. So returning back to these non-natal rears, we have this evidence that they exist, and also in more and more studies, evidence that they do survive and contribute to the adult returns. In other words, they're contributing to that salmon portfolio. So just some examples. This work by Koski et al. I really like. They emphasize that if you were to run a seine through many estuaries along the west coast at the right time of the year, you're likely to encounter these little coho fry. And those coho fry were thought for many years to be surplus production and likely to perish in the ocean. But with new technologies and the ability to track the fates of individual fish and the ability to reconstruct the life histories of the successful adults, we now know that those fry that outmigrated to the ocean or to the estuary early in life contribute, in this particular case, more than a third of the adults returning to this system, right? And these authors argue in this last sentence here that this life history contributes to this stabilizing portfolio effect by spreading the risk of reproductive failure. Later today, you'll hear some examples from Mariska Wazinski and Hank Baker describing work from the Russian river system, in which Mariska shares ideas about the factors that contribute to the expression of this early life history, and Hank discusses some of the factors that contribute to the success of those different rearing strategies, so stay tuned for that work. But for this part of the talk, I really want to emphasize that these non-natal life histories, they linger on the landscape, and when they find suitable habitat, they do well, and they contribute to the adult returns and to the stability of those returns. So keeping that in mind, it's important to recognize that the habitats that they rely on have been disproportionately lost and degraded. And this is floodplains, marsh habitat, estuaries, etc. And just one example here from the San Francisco estuary, more than 95% of the tidal marsh habitat has been lost from this system. If we shift inland, we know that a similar proportion of the freshwater emergent marsh has been lost from the system. These were the engines of productivity of this system. These were the stopover habitats that these fish relied on historically as they moved from their freshwater environment to the ocean. And more than 20 years ago, Bruce McFarland, working with NOAA here in Santa Cruz, noted that Chinook salmon were moving through the San Francisco estuary move through very quickly relative to Chinook salmon in estuaries further to the north. And he emphasizes that they likely derive less benefit from the estuary than populations further to the north. And the reason that they move through is related to this. There's simply not the habitat to support them. And so they likely reach the ocean at smaller sizes and earlier timings than they might have historically. So we know that we've lost stopover habitat. It's been disproportionately lost. We also know that river main stems have been degraded. River main stems are warming. These are some data from the South Fork of the Eel River where we do work, which emphasizes that a lot of the South Fork over the summer is in sort of relatively high temperatures, stressful conditions. But there's a lot of thermal heterogeneity that the organisms can exploit, but their ability to exploit that heterogeneity is impeded by the presence of these non-native Sacramento pike minnow. So the fact that this system is warming, is prone to disease outbreaks, is invaded by piscivores, limits the ability of fish to use the main stem as stopover habitat as well. And so just sort of thinking about the consequences of the loss of stopover habitat and degradation of main stems, what that means is that individuals that spend their first and second years of life in their natal streams and move through quickly are going to be the individuals that are favored in the contemporary riverscape. And the converse of that is that the degradation of main stems and stopover habitats has selected against these non-natal rearers. And yet, that also points us to restoration opportunities and how we might think about recovery. And here, to start this part of the talk, I wanted to highlight this correspondence with Seth Ricker, who is not here, I believe. Okay, and so Seth is emphasizing here that there's anecdotal evidence from the Bear River and the Mattole rivers that there were deep holes in the lower rivers and estuaries before the big floods in 1954 and 64. And recently they were trapping in the Bear River just above the tide. And as temperatures rose, they saw thousands of young of this year steelhead bail to the estuary, very consistent with what I showed you earlier today. And he couldn't help but go down to the estuary to see where they were, but they weren't there. And the majority of the estuary was less than 2 meters deep, it was hot and it was barren. And his sense at the time was that these animals were perishing. And he wondered if this represented the ghost of a life history strategy that no longer works. And the way that I think about this is that these animals, they're doing what they've always done, but the landscape has changed. But they're also showing us the bottleneck, and we just need to be paying attention and trying to address it. So towards that end, I want to highlight one of my favorite examples of where restoration has been used successfully to reawaken life histories. This is an example from the Salmon River in Oregon, where over a series of decades, a number of marsh rehabilitation projects involving removing levees, removing structures, resulted in the recovery of approximately 70% of that original estuarine marsh habitat. And what they found was incredible. They found that that led to the reawakening of life histories in this system. In particular, they point out that having data before and after the actions allowed them to compare life history diversity before and after that marsh restoration. And what they found in comparing those life histories is that following those restoration actions, that there was this increase in the life history diversity of both coho and Chinook salmon in the system. And for coho in particular, prior to these restoration actions, there was a single life history that spent a year in freshwater before migrating to the ocean. But following restoration of the marshes, there were 3 new life histories that were picked up, all that relied on the estuary to some degree. One were fry migrants that moved directly to the estuary following emergence from the nest. A subset of those fish then moved back upstream into tributaries to rear before going back out the following year. And then there was also a group of fall migrants that moved down to the estuary. And all 3 of these life histories that were expressed following the recovery of this marsh habitat all survive and contribute to the adult returns. It's a very hopeful message that through restoration we can reawaken these life histories. So that's the sort of final point. And I'll just leave you with the three final points here. One is that I think that dramatic responses are possible if we address the life history bottlenecks for these fry migrants, that we have overlooked their potential contributions to adult returns and to the stability of adult returns. It also suggests that we need to reorient our restoration actions. A lot of restoration is focused on tributary habitat, putting wood in streams and so on, that without necessarily realizing it has favored the natal life history. Now, I think we also need to be thinking about a suite of restoration actions that can support these non-natal life histories. And then finally, I'll say that we've been kind of working on these ideas in a kind of a multi-pronged approach. There's some empirical studies, which you'll hear about later, through this conceptual work, which I'm presenting. And then last year, I had a chance during my sabbatical to spend time in Juneau, Alaska, and to interact with Ryan Bellmore and Gabe Rossi to start thinking about how we might be able to model some of these ideas and to influence kind of management models as well. And so I'm delighted that Ryan is here with us and that you'll be able to hear from him next about that work. So thank you so much.
particularly as a federal scientist, it's really a pleasure to be here. This is my first time at SRF, and actually know a ton of people, and so it's good to see, folks like Sean and Jonny here that I don't get to see very often. I haven't seen Sean in like 5 years, and got to meet a whole host of people that I've only met virtually or talked to on the phone. So it's just been a really great meeting. And thank you, Al, for being so welcoming, and thanks to all the organizers for putting this together. So Stephanie gave a really good talk, and I think set me up really well. So I'm going to build on Stephanie's strong empirical arguments for the importance of natal rearing using a novel, what we're calling a novel foodscape modeling approach that we have been working on. When I say we, that includes Gabe and Stephanie, Avi Cortez, who's sitting back there in the audience, Holly Harris is a postdoc at our lab in Juneau, and Amy Fullerton, who's unfortunately not here today, a scientist at NOAA. So why do we need another model, right? As you all know, there's a lot of salmon models out there. I'm sure many of you are familiar and probably use some of these models. A lot of models have been used to develop to examine and predict salmon population dynamics in a variety of different contexts. And one flavor of model that I think is becoming more and more popular, and for good reason, are salmon life cycle models. These models predict survival at the sequential freshwater and marine life stages for the whole salmon life cycle. In so doing, they can be used to estimate salmon abundance and population dynamics through time, and have been really important tools for identifying bottlenecks limiting populations. So in freshwater life stages, these models generally relate survival to the quantity and quality of physical habitat conditions, primarily in natal habitats. Gabe talked about this in his intro. And they relate to things like stream flow, channel morphology, pool area, sometimes water temperature. And often what these models find or identify is that natal rearing habitat for juveniles is a primary factor limiting the abundance of populations. But, there's always a but, these models generally leave out many of the things we postulate are important for sustaining salmon from a foodscape perspective. Things like the diversity of natal and non-natal habitats across riverscapes that frequently support distinct foraging and growth opportunities that are often asynchronous. The movement of fishes, hold on, I got cool animation. The movement of fishes across those asynchronies, right? Tracking those peaks and troughs in resource abundance. And of course, the diversity of different life histories that this complement of habitats and associated foraging and growth opportunities can support. So our goal, one thing that we've been working on recently, is to develop a model to illustrate how fish movement between these natal and non-natal habitats influences salmon life history diversity and population abundance. When I say natal and non-natal, I also mean the diversity of foraging and growth opportunities that occur across those natal and non-natal habitats. So along those lines, we've been developing an individual-based model, also known as an IBM, that tracks the growth, the movement, the survival of juvenile salmon across watersheds. I'm sure many of you are familiar with the Railsback et al. In-stream model. That's also an IBM model, so it's the same sort of brand of model, although ours is admittedly a bit simpler and is operating at broader sort of watershed scales. I don't have time to go into all the details of the model, but really quickly, in the model, the growth of individual fish is a function of the seasonal regimes of food availability and water temperature, influences the bioenergetics and growth of fish, as well as competition for space and food. So growth is lower when the densities of fish are higher. Movement is a function of differences in growth potential between connected habitats, whereby fish are more likely to move if the growth potential is higher in an adjacent habitat. And survival is a function of fish size. The more you grow, bigger you get, the more likely you are to survive. And I want to stress that this is a heuristic model. Sure, you all are thinking there's a whole host of other things that influence juvenile population dynamics in freshwater habitats. We realize that, but our approach here is to start simple and build complexity as necessary. If we adapt this model to specific contexts, specific questions, we would likely include some of those complexities. So for today's talk, we're going to use this model to first examine some mechanisms by which accessible and diverse non-natal habitats promote both life history diversity and population abundance. And then, in a little case study example, illustrate how these mechanisms might support healthy salmon populations in Northern California coastal watersheds that I know nothing about. So, starting with the first objective, we first created a really like when you do modeling, right, it's often great to start with a really simple version of model first to understand the mechanisms, processes, and how they're interacting with your model. So that's what we did. We started a really simple 2-patch model. We have like one natal habitat patch and one non-natal habitat patch. And then within that really simple model, we first manipulated the size of the non-natal habitat patch, starting with a really small non-natal habitat patch and then iteratively increasing its size until we had a non-natal habitat that was substantially larger than natal habitat. And then the next thing we modified or adjusted was the regimes of temperature and food availability in the non-natal habitats. So essentially, we're moving those food and temperature regimes to create asynchronies in growth potential between natal and non-natal habitats. The peaks in growth potential are occurring at different times of the year. And then for these different simulations, we initialized the model with 1,000 individual fry, and we tracked the movement, or tracked the growth, movement, and survival of those fry for 365 days, running the model on a daily time step. And from these simulations, we came away with a few different mechanisms by which non-natal habitats benefit life history diversity and abundance of salmon. And the first mechanism is sort of the no-duh mechanism that you could get out of pretty much any salmon population model, which is that more non-natal habitat makes more fish. It's an area effect, right? So as we increase the size of the non-natal habitat, we get more juveniles still alive at the end of our 1-year simulation. So we see it start to saturate. Habitat is becoming a non-limiting factor at some point. And the mechanism here is exactly what Stephanie was talking about earlier, is that as we make non-natal habitat available and grow it, a larger portion of the population is moving into that non-natal habitat, and that's alleviating density dependence both for those fish that move, that are taking advantage of underseeded capacity in non-natal habitats, as well as the fish that stay, right? They're now growing at lower densities. They have higher growth and higher survival in the model. And again, this of course assumes that those non-natal habitats have to be connected. They have to be able to move to them, and they have to have habitat and food webs that are suitable for growth in some respect. So the second mechanism we identified from these simulations was that habitat diversity. That is, the diversity of foraging and growth opportunities within a watershed promotes life history diversity. So this figure shows growth trajectories, modeled growth trajectories. Assume like fish are emerging in April, growing for a year. And these growth trajectories are from the 1,000 fish we initialized in the model. Only a few are surviving until the end. Under a situation where the natal and non-natal habitats are exactly the same, right? The growth potential is perfectly synchronous. And in this scenario, we only get these two growth trajectories. We get this slower growth trajectory for the fish that stay in the natal habitat, and this other growth trajectory for the fish that move to the non-natal habitat. So these are two, you can see, you sort of equate these growth trajectories to two different life histories. And this red bar, which I'm going to use so we can track the change in this as we go from one simulation to the next, shows the range in fish sizes at the end of the 1-year simulation. But as we start to pull these habitats apart, creating unique conditions, unique growth potential between natal and non-natal habitats, we expand the range of opportunities. Fish are exhibiting more distinct life histories that are expressed as these different growth potentials, right? Because they're moving across the landscape in different ways. And the greater we make this asynchrony, the greater the difference, the broader range of growth trajectories or life histories that are emerging from this model. And the mechanism here, again, is that these asynchronies provide opportunities, as Stephanie said, for fishes to move between habitats, tracking foraging and growth opportunities at different times and in different ways. I think if you sort of like squint, you can, there's a life history down here, sort of this range, there's 4 different groupings of fish movement that are emerging out of this very simple model. Okay, get a drink of water. So the third and final mechanism, is that freshwater life history diversity in turn promotes population abundance. So this figure is the same one I showed you in mechanism 1, where the larger the non-natal habitat size, the more fish that you get. And this is in a situation where, again, we have perfect synchrony in conditions in the natal and non-natal habitats, and we don't have very many life histories. But as we start creating these asynchronies and we get more life history diversity, we're increasing the abundance across the range of non-natal habitat sizes. And again, the more diversity we create via differences in asynchrony, we get more life histories, we get more abundance out of the model. And so what this is suggesting, this really simple model simulation, is that life history diversity begets population abundance. And the model mechanism that's driving this is that this life history diversity, these individuals that track these favorable growth conditions across the landscape, they grow larger and they survive better, as well as the fish that stay that are at lower density dependent. So there's both of those mechanisms going on. And I'll repeat what Stephanie was saying earlier. Oftentimes we think about sort of portfolios or life history diversity, population diversity, stock diversity as something that stabilizes salmon populations through time. But what both Stephanie's empirical arguments she provided and the model are saying is that life history diversity both stabilizes populations and contributes to population productivity or abundance. Okay, so again, increasing the size and diversity of non-natal habitats increases population abundance. It does this in two different ways. One's just a simple area effect. The more non-natal habitat you have, directly increases population abundance in the model. And then the other effect is that this effect of habitat diversity. The habitat diversity promotes life history diversity, which indirectly contributes to population abundance. So let's put these mechanisms to work in a case study example, a slightly more realistic case study example to illustrate how non-natal marine habitats might support healthy salmon populations in Northern California coastal watersheds. So to do this, we parameterize the model, I'll say with like realistic-ish food and temperature regimes for natal and non-natal habitats found in Northern California coastal watersheds. And we didn't parameterize the model necessarily for any specific watershed, but it's sort of inspired by the Eel River, and the regimes of food and temperature we put in there are from the Eel River, more or less. So this model actually had 5 distinct habitat patches. The first I want to talk about is this cool tributary habitat that we call our natal habitat. And here is the temperature and food availability regimes that went into the model that influenced growth. And you can see that there's definitely some seasonality and food availability. And I think this might actually be empirical data from Gabe Rossi's dissertation. And we have a cool main stem habitat, cool upper main stem habitat, similar food availability regime, warmer in the summer. We have a warm and somewhat intermittent tributary habitat. It has a very similar temperature regime to the cool main stem, but it has pretty distinct food availability regime because it's coming intermittent during the summer. Fish are rearing in isolated pools with really low drifts, so they don't have very much food availability. And we have a warm main stem habitat, warm lower main stem habitat. You see this habitat gets like really warm for a large portion of the summer, quite a bit, about 25 degrees sometimes. And then we have an estuary habitat, which you can't really see, most of you down here, but it has a more moderate temperature regime year-round with a marine influence. And we were assuming that it has really, really high resource abundance, higher than all the other habitats, pretty much all the time, based on some preliminary evidence and sampling from the estuary in the Eel River. And I don't really know anything about the Eel River, so if these don't make sense, you can blame one of these individuals, particularly this guy. And by the way, I was Googling Gabe Rossi, to try to find a picture of the stock. You Google Gabriel Rossi, I think the other Gabriel Rossi is like an underwear model. So you find an interesting array of photos. So FYI. So the scenarios we sort of did this sort of habitat aggregation model experiment. Where we said what if we just had the cool tributary natal habitat? And what life history diversity and population abundance would emerge if we just had that one habitat? And then we iteratively added these other habitats to the model. And then every time we reran and estimated life history diversity and population abundance. Pretty simple. Oh, and by the way, this is a really sad looking coho. This is sort of this is supposed to represent coho, but we could run these types of models for steelhead, Chinook salmon actually wouldn't vary that much, just some changes in bioenergetic parameters, changes in emergence timing, things like that. So here's what happens when we just have the cool tributary habitat. We just have this, the models, all the fish in the model are converging around this one growth trajectory. There's no movement, there's no tracking anything, and we get an abundance out of the 1,000 fish, sorry, I should have said the 1,000 fish we parameterize the model with, we're getting less than 10. So that's like less than 1% fry to smolt survival if you consider these one-year smolts. When we add the cool upper mainstem habitat, you see immediately we're getting more diversity in those growth trajectories, which we're equating with life history diversity, and we're doubling fry to smolt survival. We add the warm tributary habitat, we see a little bit of a response. We add the lower mainstem, we also see a little bit of a response. Definitely more diversity in those growth trajectories. And then here's what happens when we add the estuary. And we collectively, when we get to the estuary, we have 4 times higher production in terms of fry to smolt survival than when we just had the natal habitats alone. Like, I know there's a lot of things that would potentially limit fish from fully expressing this in the real Eel River. But just an example to illustrate that non-natal rearing could be really important in these systems. And I just want to give a little example of like what's going on in the model. This is like the proportion of fish abundance by the different habitat types averaged by month. And so you can see like at the beginning there's a lot of fish in the natal habitat, and that proportion is going down through time. At the same time, the proportion, you can see this is really cool, this is the mainstem, right? Not surprising, like they're using the mainstem seasonally, and there's no use of the mainstem more or less during that like 3-month period in the summer, as we might expect. But that's important because the mainstem provides access to the estuary. They can't get to the estuary without going through the mainstem. And then here's the sort of increase in the abundance of fish in the mainstem through time. Okay, so to summarize, what these model results are showing is that accessible, it's got to be accessible, connected, non-natal habitats can contribute potentially substantially to population abundance, and that's especially true if those habitats host unique foraging and growth conditions, if there's diversity in habitats and food webs that can promote life history diversity. And I think these life cycle models we use are really important, and they're really important for identifying bottlenecks, limiting populations, but I think we have the potential for integrating Foodscape modeling into these models to understand not only that whether this life history or whether this life stage is limiting or not, but to reimagine like why it's limiting, right? How can we reimagine a landscape where we could get life history diversity and greater population productivity? And I think heuristic models like this are good exercise for thinking about the complement of habitats we need to do that. One thing that we talk about in the Foodscape paper is the idea of developing a reference foodscape, if you will, right? To reimagine what the energetic pathways and associated foraging, the patterns of foraging and growth potential are at different locations, maybe historically, using indigenous knowledge, using empirical data, using our imagination in some cases. Like what could we imagine a system used to look like? And then if we can imagine it, we could potentially model what the diversity of different life histories that can emerge from that reference foodscape. And we could compare that reference foodscape to what we have now. What are the potential life histories that are the foraging and growth opportunities that are missing, the trophic pathways, the ways that are missing, and how is that constraining the expression of life history diversity that we think promotes abundance? And so, for instance, this is more Dan Bottom's work. Like, is it estuaries? Estuaries are really big. You know, they're very productive habitats. Is it access to potentially access to these very productive estuary habitats could be a key factor. And I really like the recent example showing the fish potentially move across watersheds using different estuaries up and down coastlines too. It's really interesting. I just wanted to show you something really quick. We've been thinking about how to do these sort of reference foodscape analyses, and we created this little online simulator that's still very beta version, not meant for really sharing at this point, but you can go in and you can trace your own thermal regimes and your own food availability regimes, and then look at the diversity of growth trajectories and model population abundance under different simulations. I just want to show you a quick scenario where we said, well, this is parameterized for this toy eel river model I just showed you. What if we pulse the system with, let's say, a marine subsidy in the fall? How much would that affect life history, diversity, population abundance? Or what if we tried to represent what the main stem might have been like before pikemen were there, before climate change, before changes in channel morphology that have affected water temperature? What if we lowered water temperature in the main stem and upped resource availability in the main stem in the summer? And we look at those two responses collectively. The model is saying, well, we potentially have 42% higher productivity. It doesn't mean that's what it would really be, but I think playing out these sort of heuristic simulations really helps us think about what opportunities are missing and how we might restore them. And with that, I want to thank the whole Foodscape team, not just the authors on this talk, but there's been just a huge number of individuals contributing to this thinking over the past decade, really. And I'll leave you with this photo. These are the type of foodscapes that I work on a lot in Alaska. So this is a warm brown stream. So it's a rain-fed stream. It's got a lot of tannins. This can be like 17 degrees in the summer, merging with a glacial-fed stream. And glacial-fed streams in our riverscapes, our foodscapes, sometimes can be down to 2 or 3 degrees in the summer. Turbid, like very turbid, very cold, very warm, relatively clear, and they're changing asynchronously, providing opportunities for fish to move and grow in different ways. So, have fun. Thank you.
Please talk, John.
Thanks so much for having me. It's a great honor to be part of the session. I really appreciate the folks like Gabe who organized this session. I was a little bit personally attacked when he said that generative AI is creepy. Thanks for bringing my water. Because I created this really powerful image. That I think for the first time captures what a lot of us are thinking, which is that, you know, cold water fishes like trout and salmon, they're the polar bears of freshwater ecosystems because they're so vulnerable to climate change. And so we really want to know how they're going to respond in the future, and what's the role of food potentially in meeting this response. Not creepy at all, right? And so, you know, and this sort of figure has been really well introduced by prior 3 talks, but what it gets at is that, you know, salmonids have generally lower physiological tolerances, particularly in temperature. I don't have to tell you this. And so we're really concerned because the lower portions of watersheds are increasingly already or are forecasted to, do I got a little pointer thing in here? It's all right, I got a finger. To exceed thermal tolerances, right? So this is this figure we have from this paper. One of my specialties in science is to coin new terms that are never, ever again used. So we call this the growth regime, thinking about how temperature combines with physiological performance across the landscape and throughout the annual cycle to create these patterns of opportunity. So we can see these downstream main stem habitats, they're getting above the optimal temperatures for fish. And if we run them through a physiological model and look at kind of physiological opportunity across the year, what we can see is that in the summer, through the lens of summer, they're terrible, right? They plummet, and y'all kind of got that in your previous talks. But then at the same time, if we look at the area under the curve, and I think Stephanie really captured this with the non-natal habitat, these areas have some of the most sort of potential to contribute to production. And if you look at the perennially cold habitat in the middle, yeah, it looks great in summer, but its area under the curve is smaller. And so the point here is just that these areas are probably key to unlocking the kind of intrinsic potential of watersheds to produce fish, but they do present these risks. So a key challenge is to understand is how do fish that are in these areas like this, how do they cope with summer, this period of stress? And then in the context of this talk, what does the foodscape have to do with that? What does foraging and food availability tell us about the ability of fish to cope with these high temperatures in summer. And so what I'm going to be doing today is presenting case studies that were carried out by, that were led by my grad students, Jordan Ortega and Hannah Barrett, and what we learned about how fish respond to summer in these large rivers or main stem systems, and what food has to do and sort of diversity of responses. So I'm going to start with Hannah Barrett's work, which is in the main stem Willamette River. And so the Willamette River goes from about Eugene, that's where Ryan's from. He calls it Springtucky. He's from Springfield. From about Eugene to, like Portland. And we work in like the upper half. So Eugene to Albany, Corvallis area. I realize most of you don't know where that is, but it's the top half of it. And the whole Willamette River, the floodplain habitats there have just been greatly homogenized and deteriorated. But there's still a lot of floodplain complexity there. There's still a lot of really neat braided channels. And Stan Gregory, who's actually the predecessor of the position I'm in at OSU, he did a lot of work on this, and we kind of followed up on it. So I'm going to kind of walk you through what we've been finding. And so this is just an example of the existing floodplain heterogeneity on the Willamette, just like goes around here, I bet. It's got a lot of braiding channels. And one of the features that we're particularly interested in, so in this figure, flows going upstream. And what we refer to as alcoves. And it's areas, they're connected side channels at higher flows, but at base flows, they become disconnected at the upstream end. And so like you can see this one here, it's no longer connected here, but it's being fed by subsurface flows. Mm-hmm. And so they create these pretty big chunks of habitat. And if that, I'm not going to pretend to be a hydrologist, whatever goes down below the surface is just kind of like dark magic to me. But from what I understand from hydrologists, if it has a long flow path and spends a lot of time underwater, when it comes up, it's cold. It's similar, it's kind of analogous to groundwater. So if we look at the patterns of temperature over the summer in these different habitats, what you can see is that in the subset of those alcoves that are fed by long flow path, hyporheic flow, they're real cold during the summer. Oh, I lost my little cyan one, you can see it though. So the main stem, depending where you are, the main stem gets up to around 20 or like the lower mid-20s. And these alcoves can be up to 10 degrees cooler at their coldest points on the bottom. So they definitely are potentially providing cold water refuge for fish. And sure enough, at a subset of these alcoves, we monitored fish through the summer and did visual counts. And what you can see here, and this is certainly a subset of the total fish that are in these areas, but the numbers, they come flying up in the peak of summer when it's hottest. So this provides some evidence that fish are using these as cold water refuges. I'll just show you a video of what so we want to know, the first thing we want to do is kind of understand more about how they use them, and it's eventually going to circle back to the foodscape. So this is a cutthroat trout in one of these habitats, and you can see it's kind of murky, but that dark band is a bunch more of them in the background. And what you can see is it's not doing much, right? They're just kind of chilling. And I was talking to Gabe about this last night. He saw a similar thing in the Eel River with fish in stratified pools, right? Where they're kind of suspended off the bottom. So the first thing that we want to know is, why do they kind of seem to be holding a line? And some prior work by Carolyn Gombert and others that were interested in kind of the hydrology of these habitats, they were starting to find that the cold water on the bottom, it can be hypoxic. And so Hannah, she started going and doing vertical profiles of temperature and oxygen in a bunch of these habitats. And this is just an example. This is a lot like what you see in like limnology and showing stratification in lakes, where there's this cool zone on the bottom, but along with the cool temperatures come lower dissolved oxygen. And this is actually a good one with the measurement during the day. It's only 5, but they can get down to like 1 to 4 kind of areas. So the key point here is that as fish are looking for cold water, it's coming at the expense of low oxygen. And so there's some really cool papers from, I think it's like sometime in the last century. I forget the 70s or 90s. I don't know if it was like disco or grunge era. But they talked about the temperature oxygen squeeze, and the idea that in big reservoirs or like lakes, that fish can be forced down to get cold water, but then forced up by the hypoxic hypolimnion. And of course, but those are like, maybe places that were like 30 to 100 meters deep. These are 1 to 2 meters deep. So we were wondering, is there maybe like a smaller scale temperature oxygen squeeze? But the trick is, we tried to do like video monitoring and use VidSync and stuff like that, but it's not clear enough to actually like see how deep the fish are consistently. So then we looked into maybe like acoustic tags, but they don't have the resolution for quantifying depth. So we're like, how are we going to figure out exactly how deep these fish are. So we developed a method that we call the boat shock meat probe method. Probably another phrase I'm going to coin that will never get used again. And so we didn't actually use like a barbecue meat thermometer, but we did use a similar device that's created by ThermalWorks. They actually have like a research branch, the company that makes this. It's the top rated one on Wirecutter. But they have one that actually is a tiny, tiny little needle that's actually made for, they have ones that are small enough to be used on insects. And it's a fast response thermocouple. And so what we did is we boat shocked these big alcoves, and we could get fish like netted into hand in about like 10 seconds. And so then we would put this tiny little needle in them and get their body temperature. And then we would get a temperature depth profile, temperature depth oxygen profile, and we could map the fish onto that profile. This took, and then since I only have 20 minutes, I can't tell you about all the other things that we tried and when we cried and like failed miserably using other methods, or when I borrowed someone's some $100,000 Ditson unit and accidentally boat shocked it, but it survived. But anyways, this is the method that we found to be the only one that worked. And so I'm just going to kind of show you an example of how it works. So we had this profile, and then we look at the temperature of the fish, and we kept finding that they were mapping onto the thermocline. It was supporting the hypothesis that they were responding to this temperature-oxygen squeeze. And the thermocline depth varies across these alcove sites, and we found that the depth of fish tracked the thermocline, though there was some interesting variation that we'd love to unpack. So this supports the notion that these fish are being constrained by this temperature-oxygen squeeze. So the next question we want to know is, okay, well, do all the fish go to these alcoves? Are they critically important for this, all the population pile in here in summer? And you'd think this would be easy to answer, but again, it's kind of tricky because the main stem is too murky to just do like visual surveys. Like, we tried to snorkel it, and it's got enough visibility that you think you can do it, but then you're like flying downstream, and you can't see a log until it's like right in front of your face. And it sounds like some of y'all have experienced this, and it's like, we might die, but we're definitely not going to find fish doing this. And we spent like 8 years getting the permits, maybe not that long, to boat electrofish the main stem, but you can only do it when it's below 18 degrees C. Otherwise, we can't legally do it, right? So it makes it so we can't study fish in summer. So this is kind of zooming out. This is a big challenge in like, I think, salmon ecology is that we oftentimes like crowdsource data and then try to like quantify the thermal niche of fish and talk about upper thresholds for temperature. But if we looked at the only places that we can get like data where fish are, it's kind of biased to places like the alcoves here, right? So the way we got around this is we put temperature transmitting radio tags in the fish in spring. And I'm going to use some fancy terms here just because I learned them once. But Eulerian versus Lagrangian sampling, right? Eulerian is where you sample the habitat units versus Lagrangian is where you follow the fish. And so we did a Lagrangian approach where instead of trying to count how many fish are in the main stem, which we can't do legally, we tag the fish in spring and then we just let them tell us where they go. I probably didn't need to use that jargon, but you learn it once and you just kind of have to show off. Oh, sorry, I forgot. While I was showing off my jargon, I forgot to tell you the result of the slide. Which is that, so here's the river water temperature in the main stem, and these dots are fish. The vast majority of fish, they map right onto this, and sure enough, we found them in the main stem. So the 90% of fish stay in the main stem during summer. Only 10% move to cold water habitats, and about 5% are in the alcoves, and 5% move long distances to tributaries like the Mackenzie, where Ryan's from. Okay, so what this shows is that there's pretty extreme individual variation just in the habitat conditions that fish select over summer, right? So the fish that are going to the alcoves, they're in these habitats that are relatively cool. They're effectively zero velocity, whereas fish in the main stem are in areas that are much warmer and are high velocity. So then the question is, well, what about food? How does the Foodscape map onto that, right? And it's tricky, right? Because if you look at available food, that could kind of give you one impression. So we were sampling these alcoves, and if you scoop the kind of warm margins, they're just packed with damselflies, and it looks like it'd be great foraging. But of course, the Foodscape isn't necessarily the foraging scape, right? Like, we don't know whether those fish can access them. So what we did is we studied the diets of fish in the main stem and the alcoves, and we were able to sample main stem fish by just choosing select cool days and going out at dawn, so we could sample when it was cold enough to be legal. So what we found is that the diets were dramatically different. And so in the alcove, fish were dominated by chironomids and diving water beetles. whereas in the main stem, they were dominated by the kind of things that you think trout eat, those EPT taxa. Every once in a while you get a cool one like a Chinook salmon smolt or something, but it was mostly mayflies, caddis, and stoneflies. And then if we looked at how much diet was in there, how much energy was in their stomachs based on the weight times the energy density, we found that in the main stem, fish had over 3 times as much energy in their diet. And I think what this starts to show, and why I introduced that first part about the temperature, oxygen squeeze, is that probably mediates how the fish use the foodscape. So they're responding to those constraints, and they're kind of limited to this thermocline. And so the only things that they're probably doing is kind of opportunistically snacking on things that kind of come across that water column and across the thermocline, and that are hypoxia-tolerant taxa. That's why we think they're eating diving water beetles and Corline pupa. Okay, so Hannah did marker capture to get at the growth of those 2 different strategies, and she found that they had the same median growth rate, and they were kind of at or just above 0. So we thought, well, that's pretty cool, because one of them is eating 3 times as much food, but they're coming up with the same growth rate. So then what we did is we used just a bioenergetics approach, and we assumed that based on the diets, that consumption was 3 times as higher in the riffle as the alcove, and then based on water temperature and some other assumptions, we said, well, let's characterize our energy budgets and how they're different between the 2 strategies. And so what you see here is that consumption's 3 times higher, but costs are also 3 times higher for the riffle strategy. And so this kind of connects to some stuff that Sean's been doing in Jordan Rosenfeld's lab in Canada about pace of life theory and these kind of deep differences in life history strategies across biology. And so, in summary, there's the idea that there's these 2 fundamentally different strategies of gain maximizing versus cost minimizing. So gain maximizing, you have higher costs, but then you also have higher energy gain, whereas efficiency maximizing, you have lower gain, but you also have lower costs. And so that's a really helpful way for us to kind of understand how these 2 different strategies can have the same net outcome, even if they use totally different habitats. And it's helped us now kind of circle back. We dug in more to that temperature oxygen compression issue. And my students' animations, I don't know how to do animations. So Hannah did this cool thing where she was able to estimate fish's depth over time, like at fine temporal resolution, and it revealed this diel vertical movement that they do, where the fish move up at night as the metalimnian layer of these units becomes loses oxygen overnight due to respiration. So they kind of move up and out of that area, but they don't move up nearly as much as we thought they would. So these fish endure DO levels of like 2 to 4 milligrams in exchange for colder water. They could go a little higher to slightly warmer water, but they don't. And so those life history taxes kind of helped us understand this better, because they're cost minimizers. Their whole game is trying to cut their costs, and they don't have a lot of activity. They don't have a lot of respiration going on. So what we think is they don't need the oxygen, and since they're just trying to cut their costs to get through, that it's worth it for them to endure some hypoxia in exchange for colder temperatures. Okay, so now I'm gonna pivot to Jordan Ortega's thesis, working in the Sprague River in the Upper Klamath Basin. And I talked about the first part about this in my talk yesterday, and so I'm going to cruise through that, which is the spatial part. I'm going to talk about the temporal part, which has Foodscapes in it. So I talked about yesterday how we did a landscape survey of juvenile redband trout, and we found that they were primarily in these 3 different locations. And one was a hot, high gradient canyon, and the others were these groundwater-fed reaches higher upstream. And this is just a snapshot. So we want to know how do these 3 different areas of peak abundance, how do they actually support fish through the annual cycle? And so Jordan was out there all year round sampling fish, and I'm going to show you results from April through October. And what he found, this is temperature in the 3 different sites. You can see the springs. These are model fits, but they fit really well because the springs have a pretty constant temperature. And then here's a Chiloquin Canyon. This is daily average, but the maximum gets up to about 28 or 29 in the summer. But surprisingly, we see the weight trajectory just going up, just like it does in the other habitats. And if you look at growth rate, it's positive throughout the summer. So where this relates to the foodscape is we want to know, well, if they're able to live in this canyon where it's incredibly warm, how much more would they have to be eating? How much more must the food production be to allow them to still grow a lot and mitigate those really high metabolic costs associated with warm water? And this should be a really easy question to answer with bioenergetics. It's like what the bioenergetics model is made for. But just like you always find in science, it's like, oh, this is a lot higher than it should be. And that's because something as simple, something like Fry's like classic work on temperature and respiration, it seems like it's simple and it's just like a law of physics. And then this is a paper by Tyler and Bolduc and Tafts in 2008. And this shows different bioenergetic models fit, trying to explain the relationship between temperature and basal metabolic costs, or respiration. And what you can see is one is like this dome-shaped function, one's exponential, one's kind of flat. And we're interested in this kind of range right here. So these just are fundamentally different predictions of how metabolic costs relate to water temperature. And if you try to dig in and go, well, you know, what was the actual study like? And you go to the parameter set, you see that oftentimes they cite a different lab study, and then that lab study actually wasn't a lab study, it cites another one. And I'm not being critical of the authors because they still, like, it's just tricky. And I'm also going to show you that we just did the same thing. So we had all this uncertainty. We said, what actually is going on with respiration in this range of temperatures? So we looked, we actually did some stream-side respirometry that we talked about yesterday, but we didn't have enough resolution and temperature to really get at it. But Ken Rodnick did some really important, really cool work on interior Oregon redband trout a while back. And he has data on temperature versus respiration. And what they clearly establish is that in this kind of range that we're interested in, from like 18 to 26, there is just an increase in respiration. It's not flatlining or doming out like those previous ones show. So what we did, so based on that, then we just looked to Tony Farrell's lab, some of their recent work on warm adaptive mykiss, so that we could get an equation that we could actually put into a bioenergetics model. So we use this one right here. So we used that to kind of update the bioenergetics, and then to answer what was a simple question. I'll show you the pretty simple answer, which is that indeed, in that warm downstream reach, the fish are eating about 50% more food than they are in those colder habitats. And then what I think is interesting is if you look at the phenology of their consumption, it's actually peaking in our July sample, right? And so we oftentimes think of fish that are drift feeding in the main stem as their foraging opportunities going down in summer and peaking in spring and fall. But this is probably critical for their ability to survive in this habitat, is that somehow, maybe y'all got a grad student you're talking about who might Dyson like this, like phenology of food webs in the upper basin. But you know, somehow the opportunities are peaking as temperature's peaking, and that's probably critical for their ability to hang on in this landscape. Okay, am I over? Am I good? Okay, sweet. It says 26 minutes on here, so I was freaking out. Okay, so the key things I want to say, how do fish cope with warm summers? Well, warm riffles can support these rate maximizing strategies with big gains and big losses. Thermal refuges support cost minimizing strategies. So I want to just note that I think adults can do the cost minimizing strategy because they're not on this crazy selection pressure to get bigger and not get eaten, right? But a juvenile doesn't have a lot of energy stores. If it doesn't get bigger, it's probably going to get eaten or not survive winter. So juveniles might not have that option. And the juvenile redband trout in the sprig that are in those groundwater feeding areas, they have healthy amounts of growth. So I'm not trying to suggest that those are like refuge habitats and that they're the same as those alphas on the Willamette. And then what I want to close by is we're oftentimes asking, can salmonids eat their way out of trouble? Can high consumption, mitigate warming, and do we not have to worry? And the answer is sort of yes, but probably no. And well, this is how I think of it with like bioenergetics frameworks, which is that if we hold ration constant, as you increase temperature, like to these warm temperatures, growth starts to kind of decrease, and then it goes off a cliff. And it doesn't have to decrease. So this is the area right here where if you have the potential to increase consumption and increase growth rate. So if there's more food as it gets warm, then your trajectory could start going like this, right? This is kind of This is the range of opportunities, right? But then there's this point when you start to run out of aerobic scope and fish stop eating to protect their aerobic scope. And so basically their maximum consumption rate crashes. There's this cliff. And you can see here now the two ranges of ration, they just get pinched and ration doesn't matter anymore, right? And so at that point, adding food doesn't matter. But the thing is, we don't know where that cliff is because the more great work that physiologists do, the more we start to understand there's interspecific variation interpopulation variation, and even intra or inter-individual variation due to things like acclimation and local adaptation. So we don't know where that cliff is. So what I want to close by is saying that there's a lot of uncertainty in how consumption responds to temperature. There's a lot of uncertainty in where this cliff is, and what that means is that there's a lot of really important work still to be done on this topic. So it's kind of sad because we can't make predictions, but it's kind of cool because we all still have a lot of great work to do. And thanks so much for letting me ramble. And big thanks to my grad students, all the collaborators, funding. It's so sad it's not going to be there in the future. And then Gabriel Rossi for organizing, and also just it's so thanks. It's been so wonderful to see all the federal folks here who've been affected, and thank you for all your good work in public service.
Yeah, thanks so much for coming, and I really want to thank the organizers and especially Gabe for putting the session together and the invite to come down. Also, just want to say it's been just a really wonderful experience to be part of the Foodscape group. Over the years, I'm really glad that I answered the phone when Gabe cold called me back in 2017. I was like, California, like, but it has been just a really great experience that's shaped a lot of the thinking that I've done and work that I've done. I'm definitely an outlier at this meeting, being a researcher from Canada. And I think being down here from Canada, it's, kind of need to recognize just the extraordinary times that we're in with respect to relationships between the two countries. And I just want to acknowledge that, while it's really great to be here and connect with you all on salmon science, it's very much a privilege that I have as a dual citizen and other aspects of my identity that many other folks don't. And frankly, a lot of Canadians are quite scared across the border. And I hope that's a situation that doesn't ever become normalized. So with that said, I'm going to talk today. So with that said, I'm going to talk today about some work that we're doing applying Foodscape concepts in the interior of British Columbia. These are pretty different systems than in California, but hopefully a lot of the themes and approaches still resonate. All right, so starting at the beginning, I think the Foodscape concept in a lot of ways is based on this foundation that I think is intuitive to all of us, that watersheds are heterogeneous, dynamic, and diverse landscapes. And as we think through the lens of fish and other mobile consumers that use these landscapes, it kind of intuitively follows that some places and some times are going to be good, so they're going to be productive, they're going to be growth hotspots, I think is the term Gabe used, whereas other places are not going to be so good. So they're going to be risky, or you might have higher mortality, self-lethal effects. And so this kind of spectrum between the foodscape and the deathscape, so to speak, on the other side is a pretty simple way to look at the world, but I think underlies some really critical questions that a lot of us grapple with in watershed science and management. And those are things like where and when do we protect in watersheds, what specific process or structure we restore, and then how are these things changing with things like climate warming. So today I'm going to talk about some work coming out of a research program that I co-lead in the North Thompson. Oh wow, that map is white on the screen and not white on the slides. All right. But you can see the outline of it. And so the North Thompson is in the interior southeast BC. It's one of the major tributaries of the Fraser River, which is like the biggest system in the province. It's about 20,000 square kilometers, and it's on unceded territory of the Secwepemc First Nations. And in a lot of ways, like I said, this system's really different than California. Its hydrology is influenced by snow and glaciers. It's completely unregulated and predominantly wild populations of salmon. But in many other ways, we're dealing with a lot of the same issues that you've been dealing with down here for decades. Been increasing wildfires, droughts, and expanding human footprint. And so, like many watersheds here, there's lots of questions about, how do we steward and restore possibly these systems in a changing world? And that's really the kind of broad motivation for this work. And the focus here is on interior Fraser coho, which are a threatened population in Canada. And so really the broad objectives of this program are to first identify important coho habitats across this big watershed, how they contribute to productivity, and then also to try and better understand habitat vulnerability and the responses to climate warming and human impacts. I want to recognize a number of biologists and graduate students that have contributed to this work, and give a special shout out to Doug Braun, who's another research scientist who I co-lead a lot of these projects with. And through this program, we've tried to take a really comprehensive watershed scale approach to understanding salmon ecosystems. So really trying to think about and measure everything from the abiotic conditions of fish experience, their physical environments, the food that they eat, and then a bunch of the work on fish themselves. I won't go into detail on all these components, but these are just a few of the kind of core elements that we've been doing. And the focus here being on coho, we're really drilling into these two rearing habitats that coho use in this watershed. So one being the natal tributary streams, and then also these non-natal off-channel floodplain wetlands on the right. So you can see kind of a zoomed out aerial photo. These are almost entirely created by current or historic beaver activity and comprise a pretty large area across the watershed. And sometimes I like to pretend to be a hydrologist, and so we've done some work to try and identify flow thresholds where these seasonally connect and disconnect. And we've found for the vast majority, it's around a 1 to 1.5 year return period. So basically lots of seasonal action in terms of these things flickering on and off in terms of fish access, but some interannual variation as well. A bunch of work looking at fish habitat use, and I'll just briefly highlight this recent study from Britt Milner, who is a graduate student, and she looked at, Britt looked at coho use of these non-natal wetlands, and basically found that fish use these habitats year-round, entering during the spring, which is the big kind of snowmelt freshet, and then out-migrating either the following spring or actually in more cases than not, or more cases than you would think, getting stranded and as 2-year-olds out-migrating. And so, basically kind of setting up, you have, and connecting to Stephanie's talk and others, kind of thinking about these non-natal habitats and natal habitats as these contrasting environments that fish are using across this landscape. And that's really the key context for a lot of the rest of this work. Okay. And so a backbone of a lot of this is, that map showed up. Odd, okay. Backbone of a lot of this is a fairly extensive monitoring network that we've set up, looking at flow, temperature, dissolved oxygen, and some other parameters like conductivity around 40 sites across the watershed. And one of the really big things that has jumped out from this is just how dynamic these conditions are in time and space across this landscape. And so the North Thompson spans a pretty big latitude gradient. And so you have these systems in the north, as you get into the Rocky Mountains, or temperate rainforests, glacier-fed hydrology, and then systems in the south, which are arid, looks like you're in eastern Oregon, or as many Canadians pronounce it, Oregon, which I find really irritating. And so you can see the figure on the left is just a cumulative flow distribution of all the different tributary sites that we're measuring. And really you can just see the big variation in just like when water is coming off the mountains into these systems. We also see big interannual variation as well in terms of the timing and magnitude of the freshet, but also the summer base flow. And as, the consequence of that for these non-natal habitats is that they're quite dynamic. And so they're quite dynamic in terms of their area, and we've observed many of these habitats drying up. I think this picture on your right really exemplifies the kind of boom-bust nature of these, where this habitat that was probably several square kilometers is reduced to these sad puddles that are full of coho, and they're kind of a buffet for the many predator footprints you can see surrounding it. These wetland habitats also exhibit big swings in abiotic conditions. So this is showing hourly dissolved oxygen from 2021 to 2024. Each colored line is a different site. This is like the exact kind of figure that you're taught not to put in talks when you're a grad student. Total dog's breakfast of lines to look at. But the real key take home is that many of these systems are getting pretty low DO, kind of beyond thresholds that we know are either lethal or sublethal for fish. And a lot of this is just diurnal variation, where you have respiration at night. But in many cases, we have these like longer-term chronic hypoxia or even anoxia. And don't really see that at all in the stream environments. Similarly, we see temperature in these non-natal wetland habitats, frequently getting kind of warm and nasty in the summer. So 20 degrees is warm, and it's Canada, right? So we're not getting into the crazy 28s, but it is getting warm, kind of beyond where we think is potentially stressful for these fish. And some parallel analysis that I won't have time to talk about, suggests this is linked to the extent that these habitats are connected to the main stem river, which again flickers on and off seasonally. All right, so that's kind of the setup context, and we wanted to then, in this program, take a Foodscape lens to really think about juvenile coho salmon performance and how it varies across this landscape, and how it varies across potentially environmental variation across this landscape. So that brings us kind of back to the Foodscape concept, which we can put into this more kind of traditional bioenergetics framework, where we think about temperature as your kind of primary driver of both your kind of basal costs, but also your how much you can eat in a day. But then your realized growth, or what we're calling ecological growth potential, also being influenced by food abundance and accessibility. And I think this food abundance and accessibility piece is really one of the critical components of the Foodscape concept, also one of its biggest uncertainties, and that we don't really have great tools to predict it, and often that's kind of your limiting factor in terms of data to characterize it. So this was the basis for Sheena Parsons' master's thesis. So Sheena just defended about a month ago. She's working with me and Chelsea Little at Simon Fraser University in Vancouver. And Sheena really dug into this topic of food availability and how it varies across streams and wetlands in the North Thompson watershed. And took inspiration from this follow-up Foodscape paper that was led by Valerie Wallet and Amy Fullerton, where we expanded a bit on the original Foodscape concept and articulated these different dimensions of food availability that kind of push through these filters of physiology that ultimately determine your growth potential. And so Sheena, I did a bad job of scoping Sheena's master's thesis properly, but so she did a ton of work on all of these components, way too much work. But I'm just going to talk about two of them here. So the first is prey abundance, which we defined as the biomass concentration of invertebrates that coho would experience in these different environments. And then the second is prey accessibility. And we really like wrestled with how to unpack prey accessibility and actually quantify it. Here I'm just going to talk about it in terms of the overlap between food abundance and abiotic conditions that may be unsuitable. So kind of relating to Jonny's talk, if your temperature and oxygen are not suitable to be there, that food is probably inaccessible. So Sheena sampled 6 streams and 6 wetlands in 2022 and 2023. And the idea here was to capture the kind of seasonal trajectory of food availability that a single cohort of coho would experience in each of these habitats. So she used plankton, horizontal plankton tows in wetlands, sampled drift in streams. And basically what she found, in a nutshell, was this really striking difference between food abundance in these different habitat types. And so here, this is the invertebrate biomass concentration per cubic meter of water, with the wetlands in red and streams in blue. And basically the take home is there's like an order of magnitude difference in food availability, or food abundance, I should say, in the wetlands. The other thing that Sheena then did, it was to then match those observations of food abundance onto the abiotic conditions that occurred that day in that habitat, as just a really simple way to kind of explore whether harsh environmental conditions may be limiting food accessibility. And so this plot shows observations of food abundance with the size corresponding to how much. And it's plotting that in temperature and dissolved oxygen space. So basically, as you get lower and to the right, you're getting warmer and lower DO. And really the kind of main point here is that across all these data, we do see this trend where often the highest observations of food abundance correspond with abiotic conditions that may be getting into a stressful range for fish. And so, this suggests that, there may be this kind of trade-off between food abundance and accessibility in these habitats. Or alternatively, if fish can behaviorally kind of thermoregulate or, still exploit these areas, it suggests that these habitats where we may go out and measure temperature or DO and then conclude that they're garbage for fish may be really important for foraging. So with that work, we've arguably characterized, at least for this system, that key dimension of the foodscape, which is the food abundance and accessibility. But there's still more work that we needed to do to then kind of actually translate that into this ecological growth potential. And so I'm going to kind of finish off by just talking through the approach that we're taking to do that, which is really through the lens of individual fish, how they experience and acquire energy in different environments. This is like very preliminary and new, so any feedback or harsh criticism is very welcome. So again, we're basically arguing here that sampling in basically this lentic environment in wetlands and a lotic environment in streams is comparable when we're expressing food as a biomass concentration, right? So it's basically, the logic is that despite these different foraging modes, ultimately fish are encountering, they're scanning a given volume of water and encountering food in that volume of water. And so this initial comparison is really what a fish would encounter in a set volume of water in both environments. We need to acknowledge, though, that these environments are very different and fish experience them in different ways, and that affects how much volume they can actually search, right? And so this led me into a weird rabbit hole that I'm kind of still digging my way out of. And those, I guess, backing up for a second, how much a fish can actually scan, how much it can search in a day, it depends on a lot of things that we know are just really variable among individuals, right? So like how many hours a fish spends feeding in a day, its microhabitat selection, we know that that's really variable. But it also depends on things that like a fish's reaction distance, its swimming speed, and its capture geometry that are actually fairly conserved and that we actually have fairly well worked out. And so as a result, we can generate basically approximate distributions of search volumes within lotic and lentic habitats. And then with prey concentration, we have like the two ingredients for prey encounter rates, which is basically the total mass or number of food items that a fish encounters per day. And I'll be happy to talk about the details for this later, but all I'm really doing here is just rearranging some of the geometry in drift foraging, net energy intake models, and then taking some simple visual foraging models from Dave Beauchamp and others for fish and lentic habitats. So once we have those prey encounter rates, we can then kind of push them through capture success functions, which are pretty well worked out in streams. For the wetlands, we can use some real basic kind of disk functional response equations and plug my nose a bit. But then, we kind of end up back in our kind of cozy space, which is the Wisconsin bioenergetics model. And so, putting that all together, then we can start to model growth regimes, like I'm going to use Jonny's term in my talk. We can model growth regimes in different habitats. And so this is just a really preliminary simulation that I did where I took real temperature and DO data from just one of the streams and one of the wetlands that we're monitoring, and then worked through that process to model realized growth from mid-May to mid-August. And so don't put too much stock in the absolutes here, but it does suggest really contrasting growth regimes in those habitats. I also played around with a situation where we had hypoxia in a wetland, which we know can have sublethal effects on growth, and so you can see that effect there. You can also potentially take it a step further and envision applying thresholds of mortality. So for example, if oxygen getting below a certain threshold, as again a really simple way to just explore risk in addition to growth potential. And so that deathscape in addition to the foodscape. And so, ultimately where we'd like to head with this is to really better understand how climate and hydrological variability influences that kind of envelope of growth regimes across this foodscape. And again, this system's different in that a lot of these processes, I would argue, this watershed's fairly intact, but we're really on the front line of climate change. And so I think quantifying like that variation and that kind of envelope of growth regimes that we have now is important as we move into the future. And again, kind of trying to get at these questions of how we steward and restore these systems in a changing world. So with that, again, just like to really thank you all for coming, and happy to take any questions. And also just solidarity with federal public service.
So yeah, today I'm going to be talking about differences in young deer size between locations in Northern California. So I'm going to be comparing two different types of hydrology. But before talking about that, I just want to give a bit of context as part of a project which is looking at groundwater hydrology, feeding springs, and how that leads to resiliency of these spring ecosystems, volcanic springs that is. Really thinking about how recharge elevation, like on Mount Shasta, and changes in recharge with future climate change or prolonged droughts might affect these critical ecosystems. So, that's not what I'm going to be talking about today. Today I'm going to be talking about just differences in size. And I think it's intuitive to everyone that different locations has different sizes of organisms. A really great example of that is the Mendocino pygmy forest, where you have species like this, the lodgepole pine. Oh, sorry. Forgot about that. Where you have, too loud now? No. Where you have species like lodgepole pine, which, because of the soil conditions, they're poorly drained, it's acidic, poor nutrients, will only get about head high. This is a mature specimen here. And then you can find a lodgepole pine out in the Sierras, which is 40, 50, 60 feet tall. So a large variation in size based on your location, which is a natural variation. And we see this across organisms. We see this with parasitoid wasps, where your host can dictate your size. So a larger host might have more energy resources, and the parasitoids which emerge out of that host are larger. There's also things with host immune response, the location, the host being important. And then we also have things like lizards up in high altitudes are smaller than lizards at low altitudes because of truncated growing seasons and less resources. So there's this contrast in habitat, contrast in location. And this is a fish talk. So we have these strong contrasts in habitat in Northern California. This is primarily up by Mount Shasta that we're going to be talking about. But we have streams which are precipitation driven or runoff driven, where we have our classic watershed. We have inputs coming into a watershed. And then we have a pretty rapid hydrologic response, right? We have snowmelt maybe here, driving a big hydrologic response, and we have individual storm responses occurring over 24 hours, short time scale. And that really contrasts with volcanic spring-fed systems, where we have a watershed, and we have the same inputs. We have rain, we have snow accumulation, but it doesn't have, or it goes down into a large aquifer, right? We don't have runoff necessarily because it's volcanic material. It's incredibly porous, it absorbs everything, it eats it up. So it goes down into a huge aquifer, and over tens of years, 30, 40 years, it emerges up out of the ground. So instead of running off down through, it goes through fissures in the ground, emerges up, and you get a weird looking watershed like this, where it just comes out of a single point. And this is a picture of Rainbow Springs on the Fall River, and essentially that's it. If you were to look around it, there's no watershed upstream. Whereas this is Castle Creek near Dunsmuir, and if you look, you see mountains and your classic watershed. So, huge contrast in hydrology. And hydrology, as my Ph.D. advisor would say, is the master variable. So, it's no surprise that we get a huge difference in fish size. So this is a study done by Lissardi et al. And published 2023. And what they did is they looked at young year fish in a precipitation-driven stream, Castle Creek, and they compared that to young year fish in a spring-fed stream, Hat Creek. And what they found is the fish in a spring-fed stream were 1.5 times longer, 4.5 times heavier, fatter than those precipitation-driven stream fish. And they found that, one, these springs have a lot of food, there's a lot of food resources there, but also these fish in the spring-fed stream were a little older. So, this is a pretty new study, and we're just trying to understand, is this pattern universal? If we go out to a spring-fed stream, we look at young year fish, are they all going to be chonkers, like the one we saw in that picture before? And of course, being scientists, if so, why? So last October, we went out and we went to a series of spring-fed sites. So we have Fall River out here, got that FHH. Hat Creek, we went back there, and we went to Rising River, another spring-fed site. And we shocked up a bunch of fish, weighed and measured them, and killed them so we could extract our otoliths. And we also went to a bunch of precipitation-driven streams, which are over here to the west. Again, Castle Creek, one's in the Upper Sac, a few in the McLeod drainage. And we also have these really interesting systems, which I'm not going to talk about today, but I forgot to take the data out of this talk, so you'll see it. But we have these systems which are hybrids. So they're not only a precipitation-driven system, they're also a spring-fed system. And a classic one of that is Bernie Creek, and you can kind of see you have your watershed up here, but down low, you have a massive input of springs, and that actually makes up the discharge for most of the year. So what did we see? Well, we just found exactly what Lissardi et al. Found last time. And that is that fish in these precipitation-driven streams, this orange color down here, were pretty shrimpy in comparison to our fish from our spring-fed streams in the yellow over here. And this is mass fish length was the same story, same pattern. We also, seems it's here, our hybrid systems. Big Springs is a location on the Cloud River. Burney Creek, which I talked about, and Moss Spray is the upper sack of Moss Spray Falls. Kind of have this intermediate condition, which is really interesting. Maybe someone will ask me a question about that later. We also, so fish were larger in those spring-fed systems, but we also saw there's a lot of variation in their size, which is pretty curious. And this place in Fall River, we saw that we also had some pretty small fish as well as some very large fish. Okay, so when I was out there collecting these fish, I was like, this is awesome, this makes sense. We know that these springs are incredibly productive, so a lot of work has been done out there, and they have extremely high macrophyte abundance, and along with that extremely high macrophyte abundance, they also have extremely high invertebrate density. So maybe about 4 times the density that you'd find in a runoff stream, you'd find in the spring-fed stream. And I actually have worked in these spring-fed systems forever, since being an undergrad, so none of this surprised me. I was like, oh, of course. So what's going on? Fat fish, lots of food, probably growing pretty well, right? Well, we took the otolith data, and from the otolith data, counting the growth rings on the otolith, we could think about how old these fish are, and then think about the growth rates of these fish, these individual fish. And we're still going through all our otoliths. Like I said, we collect this data in October, and otolith preparation takes a while. So this data is all preliminary, but I have it for a few sites. So Castle Creek, this green right here, is our precipitation-driven stream. And then these other colors, the yellow, blue, and red, are our spring rivers. And I saw these results and I was really disappointed. So we see that there's not a huge difference in growth rate between these fish. There's not a statistically different or significant difference. And we look at some of our spring rivers and it's lower growth rate. Some are a little higher, some are very variable. And I thought, okay, well, that sucks. There goes this talk. And then I was like, okay, well, what about the condition of these fish? And so I was able to use all our data here and just plot simply fork length to weight. And Fall River, like I said, is really interesting. It had really small fish, really big fish, and here comes in really handy for comparison. So blue is Fall River, and you can see we got a monster, and we had a little tiny guy here. And that allows us to compare sort of like the weight condition for that fork length to our precipitation-driven streams, which is Castle, South Fork, McLeod, all these brown colors. And there's a few which drop off, but by and large, they all seem to be in sync, which is very interesting. And even some of our larger ones have about the same condition as these really productive spring-fed streams. And that was the animation to show that. So it seems like this, I talked about that density, talked about that productivity. Yeah, and I think we also, where we usually think, okay, more food, more growth, bigger fish. And maybe we're, at least at this stage in their life, more food doesn't mean more growth, or doesn't mean better condition even. This is a simple Monod type curve, and we're kind of in this land of diminishing returns. We've got a huge difference in density, but not a huge difference in growth. So, what's going on? That's a fish we caught, which is young year, and that's a fish we caught, which is young year. Huge difference. And I bet a lot of you, the answer is pretty simple. We found that there are much different ages of these fish. So our fish in Castle Creek were very young comparatively to our fish in Hat Creek or Rising River. So these fish emerged in June, whereas these fish, these big boys, emerged in March, which is quite a big difference. And we also had a spectrum of emergence in Fall River. So it seems like age is driving this difference, or this apparent differences in these. And really this comes down to this hydrology kind of facilitating different life histories between these fish, right? So we're in our classic precipitation-driven stream, and we just have this narrow window to spawn, right? You guys laughing at my hydrograph? I drew that. It goes backwards. So you have a narrow window of spawn. Otherwise you get wiped out. Whereas in our spring-fed stream, I lost my train of thought. In our spring-fed stream, conditions in terms of flows are always really good because it never really changes. And so they're able to potentially push their spawning times earlier and earlier. And what we see in Fall River, talking to Carlson Jeffrey's group, is that they may just spawn continually. Conditions are always really great for growth. And that's how you get that one. And this isn't something new for spring systems. If you look at other springs throughout the world, throughout the United States, we see this relaxation or change in life history as a pretty common feature. So we see this in amphipods, in streams in Pennsylvania, limestone streams, and basically they'll have continuous reproduction throughout the year. And if you look at springs, like Silver Springs in Florida, you see this in spotted sunfish, where they will breathe throughout the year, where in nearby tributaries or other systems, they have a distinct spawning period. So what does this mean? Like I said, I wanted this to be about Foodscapes. I saw a fat fish, and I immediately thought more food. But I need to teach us something about the role of time. So H.C. Odom, who did a lot of work in springs, really said that springs are these natural laboratories where we have variables held constant. And what we can see is that time is influencing growth overall. And it's something to think about in our foodscapes as fish are moving through these hotspots or temporally. All right. So, main takeaways from this is obviously this contrast in hydrology leads to differences in size year on year. It's not necessarily at this size that productivity seems to be governing the differences. It seems to be this difference in life history, really changing what we see there in different size. And it kind of comes back to hydrology and ecosystem stability, relaxing these life histories. So like I said, a lot of this is preliminary. We have another year to go out and collect data, and we're going to go back out in October. We're going to integrate things like temperature, maybe do some bioenergetic modeling to really see what the differences are in growth, and hopefully expand this to other rivers. All right, any questions?
Thanks for the introduction, Gabe. Thank you all for being here. It's pretty fun to have the full circle moment of getting to see some of my really early mentors in the audience that have stayed academic idols for much of my career and now get to present among you all. So today I'm going to be talking about some of my work trying to apply this sort of foodscapes lens to look at the effects of a recent wildfire on a coastal watershed located right here in Santa Cruz County. And it should come as no surprise to most of the folks in this room that salmonid populations have grown increasingly vulnerable to the effects of climate change throughout most, if not all, of their range. Most of my work has focused on juvenile steelhead populations in coastal California, which are no exception, and much of this work has focused on environmental change in the form of rising temperatures and drought on survival and growth during rearing time in freshwater. But in addition to the direct effects of drought and temperature on other salmonids, these combinations also add to a third growing concern for these populations, and that of course is wildfire. Wildfire severity has increased nearly fourfold in the last 2 decades and represents a particularly acute manifestation of climate change with potential to have both immediate and long-term effects on the landscape. However, while quite a bit of attention has focused on the effects of wildfire in terrestrial ecosystems, its effects in aquatic ecosystems has been relatively understudied until recently. Now, if we were to all start brainstorming, it would be pretty easy to come up with a pretty overwhelming list of ways in which wildfire can impact salmonid resilience. And that's because we know stream ecosystem function is inextricably linked to its surrounding landscape. Some of these potential factors can include factors in physical habitat. So we know fire can impact the physical and chemical structure of the surrounding landscape, and that can have consequences for stream morphology, substrate composition, nutrient inputs. We can also see reductions in riparian cover, which provides shade and thermal insulation for maintaining stream temperatures. It determines light availability for photosynthesis and serves as a source of allochthonous inputs in a lot of stream food webs, which means that those changes can then trigger bottom-up effects in primary and secondary productivity, as well as top-down responses in the physiological demands and feeding behavior of consumers. And all of this can ultimately affect abundance, growth, and rearing in juvenile salmonids. Clearly, this slide has gotten pretty overwhelming pretty quickly, which again is sort of the point, in that there's a lot of different ways in which wildfire can simultaneously impact these elements of the foodscape that we've been thinking about throughout this session. So there's so many different variables at play that one of the main goals of our group was first to kind of go back to the basics and think about simplifying this into a framework for assessing the impacts of wildfire on the landscape. And the way that we're thinking about this is to boil down to these variables, thinking about the effects of fire on physical habitat, which then can impact both metabolic demand and food availability in food webs that ultimately impacts survival, growth, and abundance in salmonids. So to illustrate some of the ways that this framework might play out, pop-ups. We can think about a few different scenarios using energy as sort of the shared currency for comparing supply and demand after a fire. So as a starting point, we may think about, or we can assume that food availability, if it's not a limiting factor in a system, can be represented here in green, where the energy available exceeds the amount being demanded by juvenile steelhead or salmonids. Now, we know that the magnitude and direction of these responses can vary based on the relative magnitude of this plethora of variables that are going on in response to a food fire. So in one case, oh no, I lost my, that's unfortunate. Well, there was a graph here. Did we see the graph originally? Was it in the first one of these? Okay. All right, imagine that the axes are still there. We've got a before and an after, some energy, and we've got some dots moving around. So in some cases, we could imagine that green food availability dot could decrease. This could be a situation, say, where erosion has caused sedimentation, it's eliminating habitat for benthic invertebrates, decreasing food availability. In another case, we may see an increase in light availability, and that could promote primary production and lead to an increase in food availability from the bottom up. We also have to think about some of the scenarios affecting the consumer side of this framework. There used to be dots here. Just imagine there's this really cool animated figure that I spent all night working on.
Way to adapt.
But we're adapting. At least I still have some notes. So on top of that, we could imagine that those same rising temperatures are increasing the metabolic demand of our consumers. And on top of that, this got touched on earlier, foraging becomes more costly. So we actually see this compounding increase in overall energy demand from that side of this supply-demand balance. I promise it looked really cool. Now what we're left with is a relatively simple way, even simpler than I had planned, to consider the relative effects of these ecosystem responses. And it allows us to make some kind of qualitative predictions about growth and survival for salmonids. There was a figure here too from the Rune et al. Paper that did a really nice job conceptualizing some of these in a stream landscape. And one of the ways researchers have gone about formalizing these predictions is through a variety of modeling exercises, some of which have been touched on today and applied to this kind of fire angle recently, including this really cool recent work by Rune et al. And they found essentially that in addition to these kinds of response variables, really varying in both the magnitude, direction, and duration of effect, aquatic ecosystems were particularly dependent on fire severity, time since fire, and trophic level. And those changes were largely driven by changes in water temperature, canopy cover, riparian shading, and turbidity. These sorts of studies are particularly valuable given that empirical studies of wildfire are usually pretty limited by what data happen to be available preceding this unexpected disturbance. And so opportunities to quantify ecosystem responses at any sort of relevant scale can be pretty far and few between. Wow, I lost all of my figures. That's really unfortunate. Unless, of course, you happen to be at the tail end of a PhD and still collecting more data than you know what to do with when your study system goes up in flames, in which case you are very unlucky, but also have an awesome opportunity to put some of these theories to the test. And you'll be shocked to hear that grad student was me. And near the end of my PhD, the CZU lightning complex wildfire ripped through the Santa Cruz Mountains and burned some 86,000 acres throughout the mountains, including the majority of the Scott Creek watershed, which is where I've done the majority of my research. The footprint you cannot see on the map, but Scott Creek lies pretty squarely in the middle of that wildfire footprint. And something else that was really important about this was that there was quite a bit of spatial heterogeneity in burn severity across the landscape. So we did some initial GIS analysis looking at remote sensing data that suggested that the burn severity was quite patchy throughout this landscape. And it gave us this really nice gradient to think about the effect of burn severity across this gradient at a number of monitoring sites that I had set up for my dissertation work for some earlier projects. And so that, in combination with the hordes of monitoring data I had already been collecting, set us up with this really nice natural experiment to explore these ecosystem response variables across a burn gradient. So I'm going to be sharing some of the kind of initial results from this first phase of our study, which was primarily focused on asking how wildfire affects key response variables in a salmonid food web, and identifying what some of those key variables were empirically. So we grouped them into those same kind of conceptual diagram categories that I had outlined earlier. So we had some physical habitat, food availability, metabolic demand, and size and abundance variables. And before I go into the results, I was going to show some really cool pictures of what this looked like on the landscape, some of those stark differences at some of my study sites. So this is a side-by-side looking at this same channel spanning log at one of my monitoring sites. And again, so these after photos are a year after the fire had taken place. So August 2020 was when the fire happened. It basically took over my field season, so we went out the next year, and so 2019 and 2021 are serving as our before and after time points for this sort of snapshot of the initial effects of the fire on the landscape. Here's another photo looking different direction. And now we can move on to some results and hopefully see them. So starting with physical habitat, we measured a whole variety of physical habitat variables. Some of the basics were stream depth and width. We did substrate counts, dissolved oxygen temperature, canopy cover, cobble embeddedness. We did some primary productivity assays as well. Sorry, Jonny, you don't like multivariate statistics. But we've got a PCA plot here looking at some of those variables in one space. And what we found, the blue kind of faded circle is our sites before the fire. And then the orange on the right is after the fire. And the main kind of sources of variation that we found in these physical habitat variables were, to no surprise, canopy cover. We saw different reductions in channel width and depth, and then water temperature. One of the interesting things that we found here was that our metric of burn severity, so again, this was derived from those remote sensing data as kind of a way to try to summarize overall burn severity at each of these sites. Didn't have a particularly strong effect on this like overall habitat variation response variable. And this kind of became a recurring theme throughout much of this work. So we saw a large effect of before and after, but not necessarily a huge effect of these different burn severities, which was surprising. So moving on to food availability, we did a bunch of benthic invertebrate sampling at all of our sites. And this is just kind of a coarse look at the preliminary data, but one thing we found, this is looking at benthic macroinvertebrate density. There was an increase in BMI density at the majority of our sites, and also quite a bit more variation in overall density across the landscape. Interestingly, again, burn severity did not seem to have a huge effect on predicting what the direction or magnitude of these changes were, though. The next thing we looked at was metabolic demand, and this was through a series of respirometry assays that we did in the stream. And here we found shifts in field metabolic rate that differed among the young of year and 1+ size classes that we were assaying. So this first plot is looking at the relationship between field metabolic rate on your y-axis and body size. So we know that metabolic rate should generally scale with body size, but the rate at which that relationship is scaling differed before and after the fire. And what we think was happening here was a few things. So one thing we noticed, these points, these lines intersect right at where, if you look at a length frequency diagram for the fish that we sampled, this is right where the breakoff point is between young of year and 1+ fish. And so another way to look at this is by grouping by these different size classes. And essentially what we see is we saw elevated field metabolic rates in our young of year size classes, but they were actually lower in our 1+ size classes. And what we think is happening here could be that there was some kind of selective loss of these kind of largest high octane individuals from the system after the fire. So that could be due to mortality or immigration, presumably, but that seems to be kind of what's driving this difference in the pattern there. And then last week, we did a series of electrofishing surveys to get at size and abundance. And ultimately, what we found here was that the size and density, especially of small young of year fish, actually increased at nearly all of our sites. So this is looking at fish density, again, as a function of burn severity. Again, burn severity did not do a great job of explaining the overall response, but we did see a consistent response before and after that there was an increase across almost all of our sites. And then these are just looking at the size of individuals across all of our sites, and these were taken essentially the same week of the year, 2 years apart, and across almost all of our sites, the young of year were larger in that week of sampling after the fire. So to wrap things up, I have a couple slides just to summarize a bit of what we've learned so far, and also there's a ton to unpack here still, and so I'll talk a little bit about where we're headed next. But in terms of key takeaways so far, I just want to start by emphasizing that this project adds to this, what I think is a really exciting and growing framework for using the Foodscape lens to explore the effects of environmental impacts like wildfire on salmonid ecosystems at this landscape scale. We've identified a pretty short and relatively simple list of habitat metrics that explained quite a bit of the variation we saw across habitats on fire, and those include canopy cover, stream temperature, channel width, and depth. And interestingly, we didn't find a particularly strong relationship between these variables and our remote sensing burn severity metric, which kind of highlights how we characterize burn severity on a landscape and also the scale that we're looking at matters a lot. We found some size-specific differences in how our fish response variables were affected by the fire, both in terms of metabolic demand and size and abundance, suggesting that the initial impact of the fire may play an important selective role in fish populations. And this is all consistent with much of the emergent research on this topic, but we generally found that there was a huge amount of variation in the magnitude and direction of food availability across the landscape. So this perhaps but nevertheless we saw pretty consistent increases in fish density and size, which perhaps points to this idea that landscape scale heterogeneity can play an important role in supporting overall population resilience. So a handful of next steps. Like I said, there's still a ton to unpack here. We've been collecting a ton of data in this system, and so I wanted to conclude by just highlighting a few of the visions for where we were hoping this project to head next. First, I'm really grateful to be doing this work alongside a really awesome team of scientists from the Southwest Science Center who have been continuing to monitor many of these sites so that we not only have a sense of what the immediate and short-term impacts of the wildfire were, but also get to look at some of those long-term recovery trajectories and build a time series that we can use to inform future predictions. Second, bugs. I'm currently working through a new set of community analyses to dive deeper into the fish food side of things. And those are pointing towards some cool shifts in the size structure of the benthic invertebrate community, as well as changes in the functional feeding groups we're seeing. And then finally, I'm really excited to integrate these data with some food web modeling to hopefully inform some predictions that we can make for future monitoring and recovering efforts. Thank you all for bearing with me for the technical difficulties.
Everyone, thank you so much for being here, and thank you for sticking it out. I know it's almost lunchtime, so thanks for being here. And I'm really excited to share with you some work that is part of my dissertation research at UC Davis, thinking about potential conservation applications of the microbiome, and specifically whether habitat is altering the salmon gut microbiome. So I'm really excited to share this with you because I came into grad school as a fisheries and aquatic ecologist and then kind of fell down a microbiology wormhole, and now I'm excited to come and bring what I learned back to you and hear your thoughts about how it might be helpful in our work. So to start off, an animal's microbiome consists of the fungi, bacteria, viruses, and other microbes that live on and in its body. So we, as humans, have lots of different microbiomes, and you've likely heard about especially our gut microbiome and products like probiotics or gut-healthy food. And the reason that there's so much conversation around those topics is that the microbiome can really affect host health in a variety of ways. So a suite of environmental factors like temperature, diet, interactions with other individuals and species, and the microbes that are freely available in the environment can all shape characteristics of the host's microbiome, like diversity, composition, and abundance. And then these characteristics can affect host health through a wide range of physiological functions like digestion and nutrient absorption, immune response, and even behavior. There are some really interesting studies showing that administering probiotics can affect anxiety response in mice. So the microbiome is really integral to our health in ways that we're still untangling. And I find it really interesting because it occupies this intersection between environmental conditions and health. And for that reason, I think it might be a useful lens for thinking about populations of concern. And the potential application of the microbiome in conservation has been discussed in the literature for a few years, particularly focused on directly manipulating the microbiome, so administering probiotics or antibiotics or transplanting microbiome from one individual to another. And this has shown some promising results. For example, this has been really studied in corals, and a lot of studies show that administering certain microbes to corals can inhibit pathogens, increase their heat tolerance, increase their nutrient supply, and alleviate bleaching. And so this is an area where this has been explored in a little bit more depth. But in addition to these direct manipulations of the microbiome, I'm interested in whether the microbiome could be a useful monitoring or early detection tool. So could we track the microbiome and detect early signs of change before we see larger scale impacts? Or even potentially as a habitat assessment or understanding the quality of habitat. So can we characterize the microbiome of organisms in modified habitats or reconciled habitats as another lens to understand the health benefits that those environments provide. And I've been thinking about this in the context of Chinook salmon, which we've been hearing a lot about here at the conference, a very culturally, ecologically, and economically important species that unfortunately has been in decline for many years. And I work in the Central Valley of California near Davis and Sacramento. And this decline is driven by a variety of factors, but one that I've been thinking a lot about in the context of this project is the widespread loss of wetland rearing habitat. I think this ties into a lot of the talks that we heard earlier this morning about the importance of diverse rearing habitats for salmon. And in this map, we can see on the left-hand panel in teal, historical wetland habitat, and then fast forwarding in time, almost all of that area has been converted into this pink color, which represents agricultural and other non-native land uses. And this wetland habitat used to provide really productive rearing habitat for salmon. So in the context of this widespread landscape change, salmon are experiencing a really wide range of conditions. So they might naturally rear in wetlands or in the river channel. A lot of salmon production now happens in hatcheries, and there's been some really interesting work over the past few years looking at the use of agricultural lands in these historic wetlands as sort of a proxy wetland. And I think we'll hear a lot more detail about that in talks in the afternoon session. But you might see from these pictures or understand intuitively from your experiences in these habitats that they are really different. They vary in a lot of abiotic conditions like water depth and flow, which affect temperature, which affects primary productivity, and ultimately filters up to affect the food resources that salmon experience in these habitats. So given these variation in condition, I'm curious whether these habitat changes are resulting in changes in the salmon gut microbiome. And ultimately, I'm curious about that because we have strong reason to suspect that the microbiome might affect salmon health. But before we can kind of look at that, we need some basic baseline information. We don't even know what the microbiome looks like in these organisms. So we're going to be hanging out in the middle section of the diagram here, just talking about characteristics of the salmon microbiome. What bacteria are present, whether they're changing, and if so, maybe we can describe some of those changes in more detail. In particular, I'm going to try to answer 4 questions. So 1, do salmon in different habitats have different microbiomes? We don't know yet. How much variation is there in the microbiome between individuals? Are there bacteria that are shared across all habitats? And does the diversity of the microbiome differ between these different habitats? So to answer these questions, we performed a field experiment where we took eggs from a hatchery. We raised them in the lab until they were large enough to put out in these mesh field enclosures. And then we put these enclosures in different field types. So in a managed wetland, in this case the Duck Club, in a flooded rice field, in the Sacramento River Channel, and then we kept a group in the lab for comparison. So after the salmon lived in these habitats for 4 weeks, we collected gut contents from each fish. And then we can use the DNA in those gut contents like a fingerprint to identify the bacteria. So like you could identify a person based on their fingerprint, you can identify a particular taxa of bacteria based on their DNA sequence. So this gives us a data set where we know all of the bacteria that we identified in each of our samples, and we can start to look at similarities and differences between samples and groups. So first I'm going to show an ordination plot. Sorry, Jonny. But I promise this is the only one, and all you need to know to interpret this plot is that points that are closer together are more similar to each other, and points that are farther apart are more different. Don't need to think about the axes. They aren't meaningful for our purposes here today. And in this plot, each point will represent one fish, and the color of the point will indicate the habitat that it lived in, and the shape of the point will indicate the site, because for some habitats we've had more than one site. So here come the data. Years of my dissertation work. Here it comes. Ta-da! I think we can see pretty clear clustering by habitat type. So we see that points of different colors don't overlap with one another, and this suggests that salmon reared in different habitats do have distinct microbiomes. I was really intrigued to see this result, and especially how clear it seemed to be. So I'll walk through each habitat. In the middle, in black, we have samples from the river. In orange, the samples from the rice field. In this light blue, samples from the wetland. And we can see that while none of these habitats overlap one another, they are all kind of clustered in this lower corner. And then way on the other side, we have the samples from the lab. So this suggests that salmon reared in the lab have an even more distinct microbiome from fish in any sort of field or natural habitat. I think intuitively this makes sense. The lab is a really different environment. It's very consistent. The water is really thoroughly cleaned. It's always the same temperature. The food is always the same. And so I think, to me, this makes some intuitive sense that those fish would look really different from fish that are experiencing a more dynamic field habitat and eating a more natural diet. So now that we've thought about differences between groups, we can also think about differences between individuals. And to do that, we're going to think about this plot again, but we're going to take the distance from each point to the center of its group. And I'm going to show a box plot where we have habitat on the x-axis and that distance on the y-axis. And the larger that distance, it just means individuals were more different from each other. And the lower that distance, individuals were more similar to one another. So here we see the data overlaid over that plot. And what I see is that the microbiome is more variable in the river and least variable in the lab. Again, I think that this makes sense given the conditions that fish are experiencing in these habitats. As I described already, the lab is a really homogeneous environment, so it makes sense that individuals are having a really similar experience to one another, and their microbiomes are really similar. In contrast, in the river, salmon are potentially eating a more diverse diet because there are more limited food resources, and so they're being less selective about their feeding. And there's also lots of different water inputs into the river that could be affecting the microbes that are available in the environment. One interesting consequence of this, I think, is when we think about potentially hatchery rearing and understanding that the lab is different from a hatchery environment, but maybe represents it in some ways in terms of the consistency in conditions and diet, it's possible that hatchery rearing could be homogenizing the microbiome of salmon. So we've thought about differences between habitats and between individuals, but I was also curious if there were any similarities and if there were any bacteria that we found in all habitats. So I'm going to show a plot kind of like a Venn diagram, where each circle will represent a habitat. In this case, the blue circle is representing the wetland habitat, and that number in the non-overlapping area will be the number of unique types of bacteria that we saw in that habitat. And then the overlapping regions will represent the bacteria that were shared. And then the size of the circle will be proportional to the value. So the larger the circle, the greater number of bacteria we saw. So here are all 4 habitats overlaid on one another. And the first thing that I took away from looking at this is that the majority of bacteria are unique to each individual habitat. So these non-overlapping regions circled in red are much larger than any of the overlapping regions. So we see really unique microbiomes across habitats. This makes sense with the ordination plot that we saw, where none of those habitats overlapped. But there is some overlap, and so when we zoom in on that and think about it, we see that there's a lot more overlap between field habitats than with the lab. Again, I think this is consistent with what we saw in previous plots, where the lab environment is really distinct from all of the field habitats. And what we start to see in this plot is the number of unique bacteria that we observed in each habitat, which might be starting to get you curious about diversity, and so I'm going to talk about that in a little bit more detail. I'm going to show a box plot again, where habitat is on the x-axis, and alpha diversity is on the y-axis, and then I'm going to show 3 different measures of alpha diversity. So the higher the number, the more diverse. The lower the number, the less diverse. And then as we move from left to right, we're putting more weight on evenness. So when we think about diversity, we can think about just the number of unique bacteria that we saw. So in this case, if these shapes are bacteria, in this upper left-hand panel, we just have 2. We have the circles and the triangles. So that's just our observed number of taxa. But we can also think about how even the community is and how many rare or dominant taxa we have. And so these other metrics just incorporate that idea of evenness to differing degrees. So here we see the data, and what I notice is that wetland microbiomes are the most diverse across all of these metrics, but that the magnitude of that difference and the relationship between other habitats varies a little bit when we take this into account. So particularly, we see that the river has a higher observed number of bacteria, but then it drops a little bit when we think about evenness, meaning that there is potentially a higher number of rare bacteria in that environment. I think that makes sense because we saw that individuals were more different from one another, so any given bacteria might not be observed as many times because it is in fewer individuals. So I've presented a lot of information kind of quickly here, and I'm just going to review the takeaways that I've talked about so far. First, we saw that salmon in different habitats do have distinct microbiomes. That's cool. That was kind of our core question. Now that we know that, we could also see that the microbiome is more variable in the river and less variable in the lab. That most of the bacteria that we observed were unique to each habitat, and there was relatively little overlap between habitats. And lastly, that the wetland habitat seemed to have the highest alpha diversity across metrics that we looked at. So what does any of that mean? One thing I see here is that the salmon microbiome seems highly dependent on external factors. So when we think about the microbiome, we can think about external influences, and we can also think about host influence. So in some systems, the host has some selection on their microbiome and has some control over that community. In this case, we saw an almost complete turnover in the community when individuals were exposed to a new habitat. So the microbiome seems really dependent on the environment. We also saw that these different habitats do impact the microbiome, which means that potentially these habitat changes have implications for salmon health in this new way. And lastly, what I mentioned earlier when we were thinking about individual variability and this idea that salmon that experience really consistent and homogeneous conditions, that their microbiomes also might become less diverse and more homogeneous. I also want to be clear about what we don't know from this data. So we can't really make any claim about whether one of these microbiomes is better or healthier than another. And we don't know from this data whether these differences that we observed matter for salmon fitness. So with that in mind, I want to outline a few questions that I think would be important to answer next in order to take this idea and make it a more useful tool in this context. So one thing we can think about is what functional role are these bacteria playing in the microbiome? So I talked about earlier that we used DNA kind of like a fingerprint. And just like if you used someone's fingerprint, you could identify who they are, but it doesn't tell you anything about what they do for a living or what their hobbies are, right? So we could try doing whole genome sequencing, which gives us not only the identity of the bacteria, but also potentially the function that it's playing in the community. That could start to get us closer to understanding bacteria that are beneficial for different host functions or potentially pathogenic and harmful to the host. Second, I think it's important to ask how do these changes impact salmon fitness, and we could think about fitness in a variety of metrics. So do changes in the microbiome affect their disease resistance or their thermal tolerance? I mentioned in corals and increasingly in some other taxa, there's a lot of conversation around the role of the microbiome in thermal tolerance, which I think is really relevant for salmon here in California, where we're at the southern end of the range and they're experiencing a lot of heat stress. And then third, do these early life differences have long-term impacts? So I described to you that we saw once we exposed an individual to a new habitat, within a month, the microbiome had completely changed. And so I think a really fair question following that is, well, once they start to migrate, once they go out to the ocean, does it totally change again? Do they all become more similar? Or do these early life differences have sort of persistent long-term effects? And I think it would be really interesting to pair microbiome sampling with diet or habitat reconstruction from otoliths and eye lenses in adults, and we could try to start to get at this question. So with that, I want to acknowledge that this project was a really big team effort, and a lot of people were involved in making it happen and in teaching me how to do this work. And I'm happy to take any questions.
All right. I'm here.
Talking about this.
Hang with me. All right, can everyone hear me just fine? So, Jacob Montgomery, my fantastic partner in all of this, out in the Sac Valley, got COVID, and so he can't be here. So I tried to slam some of his talk into this one. So even more arm waving and quicker than normal. We're going to cover a lot of ground because there's so much ground to cover. It is such a rich soup of ideas that we're all keying off each other right now. And I'm going to touch on a lot of them, but what I'm going to really be talking about is how Foodscapes, how energy is really at the very heart of a restoration program in the Sac Valley that is delivering economic productivity right into the heart of California's economy. So California has a $4.1 trillion GDP. So it's what? The US, China, Germany, and then right behind Germany is California's economy. And we know what this economy runs on. What? What is it? Is it salmon? No, not quite. Is it bugs? No. Water, right? It is the limiting resource. And salmon and other native fish are the primary proxy, one of the primary impediments to securely delivering that water into the economy. And the valley that I'm going to talk about, I believe fervently that rivers are much more than thin blue lines, right? But this river valley is the primary conduit delivering that mainline water into California's economy. So what we're talking about here, everything that we've been looking at in detail and in nuance kind of moves up into this political sphere. So how do Foodscapes and this pivot to process that we're in the midst of, how does that really affect the political day-to-day of the world that we live in? Do we have a clicker?
We do not have a clicker.
All right, like that, that'll work. All right, so the other thing that we have to talk about before we get going, I usually have this slide last, but I brought it up, which is the flow of fire through forests has fundamentally changed all of our lives over the last 15 years. It was always there. It's a hugely important evolutionary geomorphic process in the West. But over the last generation, half a generation, the impact of interrupting fire through not just California, but the West forests is clear to everybody in a way it never was prior, right? That capacity for human endeavor to interrupt the natural processes that create and sustain the mosaic of biophysical conditions, of the sequence of biophysical conditions that not just salmon, but the entire native biota reacts to, responds to. When those processes are interrupted, we have tremendous effects. So whether it is fire through forests or the fact that human fences interrupt the flow of grazers across grasslands, fundamentally affecting not just the capacity for ranchers to actually make money, make beef, right? With greater productivity, if you're mimicking the native herds that were once here, that natural process, you end up with greater grass productivity, you end up with rangelands better able to deliver water into aquifers to hold on to carbon better. Of course, we know about mimicking water from releases from our reservoirs, done in such a way as it was done in Pewta Creek, where instead of just saving winter's water for summer's use and creating from a Mediterranean climate, a river that had been deluge in winter and would be dry often in summer, that dynamism was interrupted with the reservoirs and water let out in a stable way. Looks much more like an eastern United States river than it does our own, and we shouldn't be surprised then when we find those critters that are adapted to those sequences doing better than the ones that we might want to be targeting, like split tail or roach. It was Jonny that didn't want roach. So all of that is just to say this idea of process-based reconciliation is inherent in all of the resource disciplines that are working. If you guys go to a holistic grazing workshop, if you go to a fire ecology workshop, you're going to hear talks that are almost identical to the ones we've been hearing this morning, except you can substitute, forest for alcove habitat, or how about the flow of bacteria into the microbiome? I mean, that doesn't just have important implications for salmon. That is at the heart of an obesity epidemic that costs the world, the globe, trillions of dollars annually. So all of this is in this idea of what I call flow theory, understanding the natural processes that create and sustain what we've often called a habitat mosaic, what I tend to now think of as a sequence of biophysical conditions. So let's try to get a little bit more specific than that arm waving. We're talking about the Sacramento Valley, where I've worked for the last 15 years or so. Before, the Sacramento Valley was one of the most productive and lucrative agricultural landscapes on Earth. It was another kind of mosaic. It was a interconnected web of river-bearing streams and adjacent wetlands. 4 million acres, a level of productivity and of diversity that is really hard to fathom, right? And of course, the Sacramento Valley is perched above the northern lobe of the Great Central Valley, so the Delta, and then the San Francisco estuary, which is what? The largest estuary on the west coast of the Americas. Estuaries are what? The most productive ecosystem we know. We're talking about a system that had unbelievable productive power that came from the interaction of water and the landscape through which that water flows. What were those patterns, right? Well, up in the Sacramento Valley, they're a little foreign to most of us who think of rivers at the bottom, usually, of a valley. In the Sacramento Valley, at least from, say, Chico, mid-Sacramento Valley, down to the Salt, the Sacramento River has built an elevated platform of its own creation, bringing the worn granite dust of the Sierra down as the river loses energy, as it tends to spread and slow. It drops that sediment, the big boulders up in the swimming grounds of the Yuba. the smaller cobble into the deltas as the rivers break into the floodplains, or onto the foothills, and then down onto the valley floor. It was the act of that water slowing and spreading and dropping its sediment that created a valley that has topsoils that are more than 40 feet deep in many places. Some places they can actually be deeper than 100 feet. So it's the very notion of this place created by these geomorphic process, the interaction of water and landscape, right? And I throw this in here. Yolo Basin is the often wetted area that you see when driving between Davis and Sacramento on Highway 80. And the point that I want to make is just, this is LiDAR. This is where Sacramento International Airport is now. This is the Feather River coming into the Sacramento coming this direction. And the only point this is up there is that the red is high ground and the green and the other bluer colors are low ground. What does that show you? The high ground is next to the rivers. Critical to understand. And why is that? Well, who was it? Was it Jonny that said the river's flowing up? How often do you find that people think water not only just flows down on a map, right? Usually south. But thinking on these simple ideas that water moves downhill. And in this case, water is going to flow during a rain event or a snow melt event in the Sacramento River and its tributaries, out over those natural levees that it had built and out into large wetlands, shallow depressions that rise gradually up to either the foothills of the Coast Range to the west or the Sierra to the east. And each one of those large wetlands could have been as much as 100,000, 120,000 acres. These are the wetlands that really define the Sacramento Valley, but they do so more than just kind of geomorphically. Those names, the Colusa Basin, the Sutter Basin, right? Those are now the names of the counties. So our political jurisdictions are based on these watershed delineations that you can barely tell unless you're riding a bicycle. The bike's a great way to get a little bit of slope out there, right? If you're driving around out there, you don't get it, but you can always feel when you're moving up a natural levee. And what happened to this amazing mosaic of wetland habitats? Well, I used to say it was channelized, but I was hanging out with Brian Kluwer the other night, and he was talking about how the English call it canalized. And it's a way better word, right? Those streams, those dynamic streams, whether they're fish-bearing main stem streams like the Sac or its major tributaries, or small creeks that run through our backyard, they've been divorced from the landscape very effectively. Often it was this simple way of piling dirt right next to every stream channel, right? But it also could be as a result of grazing or forestry land practices that lead to incision. Either way, the complete, almost ubiquitous nature of our fish-bearing streams being elevationally separated from our landscapes. In the Central Valley, in the Sacramento Valley, that is around 13,000 miles of levees that separate those once wetlands from the fish-bearing streams on either side. That's little less than 5% of those formerly wetted lands are still regularly connected to their streams, which is just a way of saying the whole, not just the whole valley, but really the whole world is ubiquitously drained. I think of humans as anti-beavers, right? We just absolutely abhor a puddle. Everything we do is to push that water off the landscape as quickly as possible. And just think about that in your life. Your backyard, a parking lot, a farm field. Everything is designed to drain quickly. So this idea that fish belong in the river and the river belongs in its banks has really permeated how, not just that we manage the rivers, but how we think about them. I mean, it definitely moves into much of the work we do too, which tends to be about that wetted area between banks, often leveed banks. And it's old as development, old as Western settlement here. This is Bishop Schofield, who was the first to go into the Sac Valley around the turn of the 20th century, really looking at things from a salmon perspective. This was just about the time that they were building the flood protection system, and he said, great, let's separate those wetlands from the river because that'll save a lot of fish. And the idea that fish belong in the river and the river belongs in its banks has really been here from the beginning of our understanding of how rivers should be managed. Yet, when you have a really steep slope, a steep transition between water and the landscape through which that water flows, it means you can put a lot more water into that canal and get very little change in the biophysical condition. Very little change in the habitat value associated. And yet, what we'll find if you look at the major investments in California in water and habitat, it tends to be about putting water into a deeply broken system. So how do we get out of this? How do we get out of this? Well, the story starts about 20 years ago on a little-known river called the Cosumnes, which flows into the, well, I don't know if starts is the right word, right? None of these stories start. But my story starts today on the Cosumnes, where my good friend Carlson Jeffries did his master's. The Cosumnes is small enough that it doesn't have a major dam. What does that mean? It means when it rains, the river comes up. And when the river comes up, sometimes it spills through levees. And in 1997, it spilled through a farm levee and put a splay of sand onto 28 acres of tomatoes that the farmer didn't want to fix. It was too much of a pain in the ass. So he moved the levee back. And those 28 acres were exposed to a river that came up and dropped back down when it rained. And within a couple years, there was willows and oaks. And it caught people's attention, caught Jeff Mount's attention, caught Peter Moyle's attention. And Peter said, well, I wonder what's happening out there for fish. And his master student, Carlson, went out and caged some fish. He put them out here in the swamp, in the wetland on the other side of the levee, where fish don't belong, right? And he put them where they did belong, in the stream. And what he found is now famous to all of us, right? This first glimpse of what a Sacramento salmon actually should be, of what a Sacramento salmon is when you re-expose it to something like, to a gross approximation of the landscape that it is adapted to, that it evolved with and under. But there's a really important point here. I already said it. There's 28 acres. There was millions of acres like that. What do? I mean, we all get this feeling, right? What can you do as a postage stamp? And so what we started to understand was, while we might not be going back, while we're not going to willy-nilly move levees that cost now $25 million a mile to move, while the ground on the other side of those levees is some of the most expensive ag land in the world, there really are possibilities. What are those? Well, it turns out, and again, this is a simple graphic of the Sacramento Valley. This is the Sutter Buttes in the middle, often called the smallest mountain range in the world. In reality, the last upthrust of the Cascade Range. And all those different pastel colors are just different kinds of wetlands. That was pre-Western development. That's today, right? There's nothing particularly noteworthy. That's what everything looks like. But in the Sac Valley, there are 500,000 acres of rice grown on those really deep clay soils that formed at the bottom of the flood basins. And what does that mean? Why is rice important? Well, rice is one of the few things where you actually make a puddle on purpose, right? So in that managed agricultural habitat, something had happened in the previous decade to Carlson's experiment that really mattered. And that was that the California legislature had banned the burning of rice stubble. Rice stubble, when I was a kid, the Sac Valley sky started in mid-July, would turn black. There was an epidemic of asthma. And clean air legislation clamped down. The legislature passed the Rice Residue Burning Reduction Act. Had nothing to do with animals, had nothing, well, besides our children. But it wasn't meant as an environmental, as a natural resource thing at all. But lo and behold, those ducks and geese that had flown from the Arctic down to the heart of the Pacific Flyway for generation after generation, when those rice fields, which had been kept more or less dry, in winter, the opposite of what it would have been historically. Now they were flooded in order to break down the silica in the rice straw so that the farmers could prepare their fields for planting the next spring. And those birds said, oh, I know what to do with that, right? That's a wetland. And they landed in those puddles, and there we saw, through the years that I was growing up in the valley, this incredible story. Birds were at all-time lows when I was a kid. Something similar to where we find our salmonids now in the state. 30 years later, they're really at all-time highs. Not all-time highs compared to what it would have been pre-development, but since we've been keeping count, we have thunderous herd, what are the herds? Flying herds, schools? No, what are they? Flocks. I should probably flock together of geese in the watershed. And to watch 300,000 geese fly over you at dusk is absolutely and totally awe-inspiring, right? And why did it happen? Well, here's an 1888 picture by William Hammond Hall, the first state engineer. Again, here's the Sutter Buttes. And this shading you see there represents two of the largest of those floodplains I was talking about. The Sutter on this side and the Colusa on the other. And what I've done is overlaid that with modern Google imagery. And it's not great in here, but you can see that 5 o'clock shadow. You can tell from space where those marshes still were. Why is that important? It's because the floodplain's still there. What changed is how we manage it. What changed is the pattern of water flowing across that landscape, right? So what can we do? Well, if we, and this was the idea, if we approximate the natural sequences of biophysical condition that these native biota know, that they recognize, could we see a similar explosion in abundance for fish that we had seen with birds? And that's what we went to test. And this is not, play this is where it's looking at. So, we didn't have large-scale restored marshes to work on, but there was 500,000 acres of rice fields. And these rice fields had been shown to be able to create really enhanced conditions for birds. This was a field that we started working in several years after our first experiment. But under all those geese are tens of thousands of juvenile Chinook. Let's talk a little bit more about how that happens, right? So in the early 2000s, we wanted to go out and see if you could take a corner of a rice field and produce wetland-like habitat for fish. And lo and behold, those fish, when they were thrown into what really looks like a muddy puddle, not only did they not die, but they doubled their weight every week. So that we started to see a pattern. And we kept testing it. We said, okay, so what happens if you put caged fish in the Sacramento River under spate, where this is as good as it gets to be an out-migrating young salmon. Each one of these cages has 10 pit tag fish in it. And then you go 30 yards over the other way into the canal. Canals are how water flows across the floodplain in a drained, plumbed, large agricultural river valley the world over. And then you just go 30 yards more onto the floodplain surface where we are using rice fields to have long duration, shallow inundation. And here we saw 3 weeks of growth. We had to use hatchery fish because they'll never permit us with wild fish. These are hatchery fish. You have to take my word for it that they grew almost not at all. These guys, you're starting to see they're getting a little pooch. But in just 3 weeks, the ones out on the floodplain looked like those. And the reason was why? Well, a cubic meter of water in the sac has about 1,500 invertebrates, mostly small. The canal, holy shit, 600 times more? That seems like that's a lot, right? But move over here. 150x, that's 15,000% more available food density. This is not anomalous. We have, in the previous 12 years now,
looked at every single kind of what I will call aquatic habitat, what everyone else just said, it was wet, right? Whether it was a puddle or a managed duck club or any other kind of aquatic habitat. And what we found was a really similar density of invertebrates. Now, sometimes there's zooplankton, sometimes, depending on the donor water, they can be river invertebrates. If that water's coming out of the feather, they can, the fish can be eating mayflies. But a very simple story was emerging, that when the fish-bearing channel spreads and slows, it accesses stored solar energy kept out on the terrestrial surface, right? What is that? Those leaves, those are batteries, right? So aquatic bioproductivity, what does that mean? It means bacteria, microbes have access to detrital and vegetative carbon that moves up very quickly, turning into dense zooplankton, and then small fish rapidly become big fish. But as rapid as it is, it does take time. It's about a 3-week process. So why is this critical? And here's a piece that I was really appreciating Nick talking about time. Water in the river turns over every couple seconds. Water in the canal turns over every couple minutes. Water in a field turns over every couple days. And historically, those hundreds of thousands, those millions of acres of wetlands could have been inundated for weeks, months, sometimes even perennially. We're talking about a fundamental shift in the capacity for a river ecosystem to actually harvest energy. It's not just extend. It's not just get it wet, it's keep it wet. It's the wetted acre day. It's the means of actually transferring stored solar energy into a river so that you can have abundance, right? That's what abundance looks like. So we went to check that out. I can't tell you the details on all of this and still cover through this stuff, but we did big floodplain laboratories that had different residence times. Everything else held the same, different amount of time that the water takes to transit a otherwise same wetland environment. And you see the growth difference. Everything started the same. You have about 1.5 times the growth, and that was us just trying to get to 1.5 hours. It's really hard to hold on to water in that. Okay? But look at that, the difference. I mean, you can't even tell the difference if you look. It's just that they just look like little fields. But if you slow down the water, you allow the microbial activity enough time to process the carbon, and it goes primary, secondary, tertiary, you start making fish. Why is that important? Fat fish, I love hearing Ryan just say, bigger fish survive better. You would be surprised how controversial that is within the Department of Fish and Wildlife. My dear friends, Rachel Johnson and Carson Jeffries, along with Miranda Bell Tilcock, are using eye lenses to actually look at habitat-specific growth rates, and I'll just do this very quick because I'm almost out of time. But the fall run, yellow is the juveniles leaving. So if you look at their eye lenses, you can tell if they ate from a floodplain-derived food web or not. Very few of them did so. The adult survivors, well more than half. And if you look at winter run, it's far more dramatic. We just got returns from the 2022 year. 299 out of 300 adult surviving spawners for winter run had floodplain signals in their eyes, despite almost no opportunity to actually get out there on the floodplain. So all of this is really translates to the politics of the valley. And 1 minute, you said? And Jacob, yeah, we're gonna. So this, everybody knows things like this. There used to be more fish, now there's not. This is spring run in the Sac Valley, but there is an anomalous outlier, and that's the small stream that runs right along the Sutter Buttes. It's called Butte Creek. So the buttes have orchards on one side, row crops on the other, but to the west, they have a landscape scale wetland called the Sutter Buttes. It's where people go to shoot ducks, and then that water moves down through a flood conveyance system called Sutter Bypass that doesn't look much like a functioning floodplain, but it is. So this environment is a swim-through wetland. The only one that exists in the entire valley. The only place where a fish could actually swim out of its stream into the productive wetland type environments and then move back into the stream on its way out. And this is the agricultural environment that feeds it. And when a couple of diversion dams got taken out, we had this amazing thing happen. From almost nothing to the incredible exuberance that salmon populations can and do exhibit when they are given every link in the habitat chain. When the landscape can provision the biophysical conditions that each life stage needs to matriculate to the next. That's why salmon are famous. They do those two things. They're incredibly exuberant, they're abundant, and they move. They synergize the system. So, beautiful pictures of Allen's showing what that looks like. What I would like to leave you with is what's happening with this. So Floodplain Forward is the political coalition that we have built around the simple idea that fish need to eat. And that to eat, you need to reconnect rivers to wetland type habitats. And we have brought together all of the largest farms, all of the ag interests like the California Rice Commission, the water districts that supply water to those ag interests, the bird and the fish conservationists, and then the regulators which work with us all. And it's all been around this simple idea that spreading and slowing water has a myriad of benefits. We're talking about puddles being the primary antidote to food security, water security, and extinction in freshwater systems. Why? Well, it's simple. You start to approximate the biophysical conditions that emerge as water moves through the landscape through which it flows. And to get everyone bought into this, it's around a simple thing. It's more profitable. When you align your ecological and your economic outcomes, you have a fundamentally different set of relationships which accrue, right? And so we have now created a $2.5 billion portfolio within the Sac Valley over the next 5 years to get to truly landscape scale. And this idea of advancing floodplain reactivation was the talk that Jacob Montgomery was supposed to give. And I'm going to take one moment to explain how farming and how fish and farms can really work together here. Not just in building large swim-through wetlands. That's the Nagiri project. That's what I've been working on. I'm going to blow right past that, except to let you show the making of a floodplain fatty. So we know that very quickly a fish that actually gets out into wetland-type habitats grows very quickly, right? And now through those islands, as we know that they're accruing the very set of physical conditions that allow to returning, abundance. That's great. It's still really hard to get fish out of those levees onto the landscape. 98% of the former landscape, I don't have time to do this one. We're going right through. The idea here is you got to, on those big landscape scale projects, the Nagiri project's 2,500 acres, you don't just get fish out there, you actually have to hold onto the water long enough to make the food and let the fish eat the food. That's what this is about, increasing the number of wetted acre days. There's also a string of similar projects that we're doing along the 300 miles from Redding to the salt, looking at remnant floodplain within the levees and how we can use cutting edge mapping to talk about not just what exists now, but what could exist if we actually hold on to water and recreate the ecological function, if not the actual historical conditions. But what I wanted to say before leaving, because Jacob's not here, he's in bed with COVID, was the fish food program that he's put the last decade into. And this just realizes that the vast majority of that wetland, that productive surface, is on the other side of levees, and fish are unlikely to be able to get there anytime soon. But it doesn't mean that that ground can't have a very important part to play in re-energizing the river ecosystem. And that's because we're working at a truly landscape scale. This was an experiment that we ran that was 7,500 acres. Those acres were flooded bugs grew in there within 3 weeks, and then those were drained continuously over a couple months, and we did similar fish growth studies looking at fish growth above that subsidy site and below. And what we saw, I guess I don't even have that picture in there, right? But what we saw was that fish as much as 6 miles downstream were growing 3 or 4 times the length of fish that didn't have access to that food. And this just simple idea of energy moving through the landscape interrupted by human endeavor. And so if we can define habitat not as a place, but as that spatiotemporal pattern or sequence of patterns of biophysical conditions that arise as water flows through the landscape, then we have a chance to really do this thing. You can't see any of that. But not just go from ridgetop to river mouth in full life history, but to define the life cycle and then understand the niche cycle, which is characterizing the ecological functions required for each life stage to matriculate. And then you get a habitat cycle, you have a process cycle, and you can start to look at where those processes are interrupted by human endeavor. What we're talking about is kind of turning our concept of what a rivershed is, at least from a recovery sense, on its head, so that we can get to this simple idea that we don't manage salmon. We don't even really manage the rivers on which those salmon depend. What we manage is human behavior. And what we have a chance to do is start to align that human behavior with an understanding of the natural processes that create and sustain that mosaic of habitat that we all know is so important. So, thank you.
Thanks to all of you folks who came from far away. It's just really exciting to get to have this exchange. So the study that I'm going to talk about today is building on some monitoring that I've been a part of in the Russian River watershed for about 20 years now. And it's also part of my PhD work at UC Berkeley. And I'd just like to acknowledge my co-authors, Stephanie and Ted, are my advisors at UC Berkeley, and then Greg Horton at Sonoma Water. I'd also just really like to acknowledge the many, many field staff and hatchery crews that really collected this massive data set and made this kind of analysis possible. Some of you are in the room, so thank you. So my talk is going to have a couple different parts. I'm going to first talk about some analysis that we've been doing to look at early emigration in tributaries of the Russian River. And then I'll touch on some foodscapes and how this early emigration work, along with some other observations, are helping us just think about recovery in a foodscape context. So just starting with some basics in the Russian, coho salmon have typically a 2 or 3 year life cycle, about 1.5 years spent in freshwater habitat, and then a few months to another 1.5 years in the ocean. I'm going to touch on some concepts that Gabe and Stephanie and Ryan talked about earlier. I'll probably be beating you over the head with them a little bit more. I've heard that you have to hear things about 7 times to just even begin to comprehend them. So just starting with, our earlier understanding was that, juvenile coho salmon were primarily living in natal habitat until the smolt stage, and then migrated quickly through the estuary to the ocean at age 1. But many studies, as we all have been reading over the last few years, have shown that there's much more complexity in juvenile rearing. Fish are leaving the natal habitat earlier, residing in the main stem and estuaries and non-natal tributaries. Now this recent paper that just came out showing that they're leaving, freshwater habitat, swimming through the ocean into other freshwater habitats. So much more complexity than we thought. And this is important because if all of the juveniles stay in their natal habitat and something bad happens there, you can wipe out a cohort really quickly. Whereas if the population spreads out across the landscape, it's kind of a way of that hedging against uncertain conditions. And it's also really important for increasing abundance. If some of the fish leave the natal habitat, there's more resources left for the fish that stay, and then there's just a much greater expanse of habitat and food availability and growth opportunity in those non-natal habitats. So all of this got us thinking about, what's happening at the southern extent of the range where there might be different constraints on early emigration, what's happening in the Russian River, what factors explain that emigration timing, and do any juveniles that emigrate early, do they contribute to the adult return population? Our work took place in the Russian River watershed. This is home to steelhead, coho, and Chinook. The focus today is on coho, which reside in the lower part of the basin. And it's a small remnant wild population of coho that's very much supported by a conservation hatchery. Also, just of note, it's a rainfall-dominated hydrograph, and intermittency is pretty typical in the tributaries during the summer dry season. So our study fish were hatchery coho juveniles that were released each year into these 4 tributaries. Sorry, I'm getting over cold. And they were, a few thousand were stocked into each of these tributaries over an 11-year period, and they were from genetically similar family groups. So a total of a little over 89,000 fish, pit-tagged fish released. So about 15% of them were pit-tagged. And that allowed us to track their immigration timing at these monitoring stations. Each monitoring station was made up of a pair of pit antennas that we operated year-round, and then smolt traps during the spring season. So our first question was, just what are we seeing in these southern systems in terms of timing of emigration? So this is emigration timing just averaged across all of the streams in all years. And this is showing between late October and the end of June. The rest of the year, the streams are dry, so there's no movement. And we did see kind of 2 pulses, the typical pattern of smolts leaving at age 1 in the spring, but then we also saw this pulse of fish leaving in the fall and winter. And we, just for the purposes of our analysis, we used a finite Gaussian mixture model to identify a threshold date that would separate these 2 modes, and that was March 3rd. So we defined early immigrants as fish leaving before March 3rd. And these are probability density plots of just the timing across all of the streams and all of the years. And this is just raw antenna detection data, and we saw some pretty striking differences between some of these watersheds. In Willow and Green Valley, very few fish were leaving early. That line is the March 3rd cutoff. And then in Dutchbill and Mill, it was much higher and variable across years. So we wanted to take into account antenna efficiency, because that can be variable across years and seasons. And we also just wanted to take advantage of these 89,000 individual detection histories that we had. And so we used individual-based mark recapture models, specifically a multi-state emigration model, to estimate the probability of emigrating early from each of these streams in each of the years. And this really kind of mirrors the raw antenna data, very low probability of early immigration from Willow and Green Valley, and higher and more variable from Dutchville and Mill. So what explains this? These were the same genetic family groups going out into 4 different watersheds. So why did we see such big differences across watersheds and across years? We were interested in looking at some different kind of higher level factors based on other studies, geomorphic factors. We were specifically interested in floodplain habitat in each of the watersheds, flow and temperature, as well as, are there any density dependent processes happening, and then, or are there size differences? And we use the same multi-state immigration modeling framework to construct models with different covariates so that we could evaluate the relative influence of these different factors on early immigration. So just a little more about the covariates we ended up using. One of them was valley bottom area. So our thinking was that if a watershed had a lot of this low gradient floodplain habitat with potentially high food availability and growth opportunity, refuge from high flows in winter, that there would be a lower probability of early immigration. And so we did this GIS exercise of calculating valley bottom area and then standardizing by watershed area. And we saw pretty big differences here. So 2 of the watersheds had relatively higher valley bottom area than the other 2. We treated this as a categorical variable. And then we were also, based on other studies, interested in flow and temperature, which can influence movement. For flow, we were more interested in flow as an opportunity for fish to express an early immigration strategy rather than just high extreme flows blasting fish out of a system. So we were interested in the magnitude of flow, but also the duration of flow. So we just calculated the days during that winter period that flow was above the data set median. And then we were also interested in temperature. We just kind of boiled that down to the mean temperature during that winter period between October and March. And then for density, we estimated juvenile densities in the fall. So that was coho and steelhead, wild and hatchery fish. And then we had individual sizes at the time of tagging. And then there was one interaction we were really interested in, and that was between valley bottom area and flow. And we were thinking that in a system that has a lot of floodplain habitat, increases in flow could actually increase the amount of suitable overwinter rearing habitat. And so that would be really different than in a system that was more high gradient and not a lot of floodplain habitat, be more of a fire hose effect and just higher early immigration with increased flows. So we tested for collinearity and only included variables that were not highly correlated. Z-scored everything, and then tested a number of covariate models and just compared them, and ultimately found that the most complex model that included all of our covariates and the interaction had the greatest support. And just looking at the beta coefficients of the top model, we found that floodplain habitat, or the VBA covariate, had a much stronger effect than any of the other covariates, and it was a negative effect. So as we might expect, higher amounts of floodplain habitat meant less early immigration. But some of these other factors were important too. This is just zooming in a little bit, and we saw that flow and density both had strong positive effects on early emigration. Temperature was less important, but also a positive effect. And size was a positive effect as well, which is a little different than other studies. So we're still kind of puzzling over that one. And then the interaction was, as we kind of predicted, flow had a much stronger effect on early emigration in systems that had less floodplain habitat. So our last question was about whether early emigrants contribute to adult returns. It's an endangered population. We don't see many adult returns, but over the 11 years, we detected 155 adults coming back to the watershed. And of those, 15% were early immigrants. So we compared the proportion of early immigrants at the adult stage to what we observed at the juvenile stage. And that was pretty similar when we aggregated across all streams and years. But when we broke it out by sub-watershed, we start seeing some differences. In Willow, the proportions were similar. In Dutchville, the early immigrants were underrepresented at the adult stage when compared to the juvenile proportions. And then in Green Valley, the early immigrants were overrepresented, same in Mill Creek. In Dutchville and Green Valley, the results were significant based on a chi-squared test. So this all just begs a whole nother series of questions. You know, what's happening to these early immigrants when they leave these tributaries, and why are some of them more successful than others depending on watershed, and how can we support you know, systems like fish leaving systems like Dutchville to help allow them to survive better to the adult stage. And then we also broke down some of our adult return data by year, by cohort, and we saw some variation there. So for some cohorts, we saw no adults returning, and others, there were a lot of adult returns. And so this also just is a whole nother direction for us to go in. You know, why are early immigrants more successful with some cohorts than others? And just one hypothesis is just that early immigrants may have a much more important role in years when we see stream flow disconnection really early in the season. So in the 20 years that I've been working in this watershed, the streams almost always disconnect, but it's often in June or July after the small run. But in recent drought years, we're seeing disconnection as early as April. And if you think about that timing of disconnection when overlaid with the average timing of juvenile emigration, that's gonna present a real problem for all those late migrating smolts. And in our system, there's massive stream drying in the summer, so it's not like fish can just residualize and go out the next year. We actually see very few 2-year-old smolts going out, and we think that they don't have much of a chance of surviving because of the summer conditions. So yeah, in these kinds of years, we think that the early immigrants are really the fish that have a chance of surviving. So that concludes part of my talk that's about the study that we've been doing. I'm going to shift gears now and talk a little bit about just how a Foodscape perspective can help us just think about salmon recovery in the Russian. Just a little background, the Russian population was nearly extirpated from the watershed in the early 2000s, but as a result of a lot of habitat restoration work that has happened in the small tributaries and the onset of a conservation hatchery program, the numbers are now averaging a few hundred per year, and that's all encouraging, but we're very far from our delisting targets. We're even farther from the tens of thousands that once returned to the Russian. And just to come back to Stephanie's question, you know, even if these natal habitats were pristine, how did they make it work? How did the populations ever get to levels that high? And again, going back to this idea of non-natal habitat as a way to really increase production. If the population can make use of these non-natal habitats, that's kind of one way you can envision a lot higher abundance. So in the Russian, this is a map of the lower coho part of the basin. The dark blue lines are the natal coho habitat, the spawning habitat. It's really been the focus of like almost all of our restoration work. It's been the focus of hatchery releases. It's been the focus of most of our monitoring. But what about all of this? You know, we haven't paid as much attention to all of this low gradient floodplain habitat that is very, very likely supported much greater abundance of these populations. And we're seeing evidence that fish are trying to use it. The early emigration study that I talked about, we're starting to see higher levels of spawning in some of our systems, and in years that we do, we're starting to see fry emigration to the estuaries, a whole nother life history strategy that we hadn't seen before. And really anytime anyone throws an antenna on one of these habitats or does some sampling, it's not uncommon to just come across some juvenile salmonids. So I think it's time to really, you know, all of the work in the natal habitat is really important, but we need to start thinking about the bigger picture and, you know, just reiterating some points that have already been made. And in the Russian, there's also evidence of higher growth opportunity in some of these non-natal rearing habitats. I didn't have coho data, but this is some juvenile steelhead growth data in a small natal stream as compared to a larger tributary in the estuary. And so really big differences there. One of the problems, though, the challenges, is that these low gradient habitats are also popular places for urban development, loss of, you know, we've just seen loss of habitat, loss of connectivity, habitat quality, you name it. And I do just want to end on a positive note. I think things are changing. There are some really great projects out there that are starting to really think about non-natal habitat and connectivity. And I wanted to just highlight these two watersheds and some really exciting work that Goldridge has been leading with a lot of funding from NOAA. Some of our key NOAA colleagues that weren't able to be here that have been really involved in this work. And there's many other partners. Goldridge just taken the lead, but increasing connectivity to floodplain habitat, increasing the amount of floodplain habitat. In some systems, there is a lot of physical habitat and there's connectivity, but and fish, we're seeing fish trying to use those habitats, but there's serious water quality issues. Fish go into this habitat and don't come out, and we think a lot of that is because of water quality. So thinking about that as well, you know, there might be food, but is it accessible? And then just one last watershed that those of us, some of us at Berkeley and California Sea Grant have been involved with some monitoring support. There's this tremendous effort to try to restore non-natal habitat in this watershed, Willow Creek. It was channelized and pasteurized and logged, and it comes in right near the estuary. So there's a huge effort by some really dedicated people to try to improve that habitat. So that's a really exciting project happening too. Okay, and I think I am out of time, so yeah, thank you.
Yeah, so thanks to everyone for sticking around. It's really nice outside, and we've heard a lot of talks today, so appreciate you coming to listen to my talk. My name's Hank. I am not a salmon person for my whole life, so I'm relatively new to the salmon scene and very much enjoying it. Longtime fan, first-time participant. So anyway, yeah, I am really interested in sort of the causes and consequences of intraspecific variation. And now for my postdoc with Stephanie Carlson's group and Ted Grantham's group at UC Berkeley, I'm starting to apply that to some of the ideas that you just heard from Mariska in Willow Creek. And so I'm presenting on behalf of Mariska, Ted, who's in our competing session right now, and Stephanie, who are my mentors and co-conspirators at Berkeley. Okay, so I am gonna just bonk you over the head with things that you've already heard today, but as they say, repetition builds learning, and repetition builds learning. So, tough crowd. So there's sort of 4 key takeaways that I want to convey today, and some of these are going to be like explicit through my talk, and some of them are just going to be kind of like background ideas. The first is that juvenile coho salmon are more diverse than is typically appreciated. I know that probably all of you in this room who are interested in Foodscapes understand that juvenile salmon do a lot more than sort of this textbook version that we're used to, but I'm just going to talk about a few of those ways that we've identified through our research. And that even subtle diversity can have large effects on population dynamics. I'm not actually sure that subtle is the right word here, but maybe fine scale diversity or small spatial scale diversity is a better term. And next, that managing for that diversity can explicitly improve conservation outcomes. And finally, that I think a lot of us have known for a long time that salmon do things that are not sort of this canonical pattern of natal rearing and then smolting and swimming out to sea. And it's present in stories that people tell in indigenous knowledge, in even like old papers and things where people are like, oh yeah, there's these fish doing all these things. But it never made it into sort of these like textbook models that we have, in part because it's really hard to make robust inferences about uncommon things. So you really need large data sets in order to draw robust inferences about these uncommon things. And so now with data sets like the one that Mariska has been working to build for 20 years now, we're able to sort of identify these and talk about them in sort of more quantitative ways and account for their contributions to salmon populations. So again, here's a typical coho salmon life history. I'm not going to go over this. You all know what this is. There is much more variation at virtually every single one of these steps than is sort of appreciated in the in textbooks. I'm going to be talking about variation in their juvenile life stages within river networks. And I'm going to be doing sort of a 2-talk presentation here. So the first part is going to be about variation in movement within a single creek. So this is that sort of subtle, or maybe not so subtle, it's fairly striking variation, but at a relatively small spatial scale, and how that can have pretty profound effects on population dynamics. And then part 2, I'm going to be talking about some new work, some sort of in-progress work about zooming out at the riverscape scale and looking at variation in non-natal habitat use and sort of trying to identify features of potential recipient habitats that are attracting non-natal rearers and also affecting the phenology of habitat use. Again, just these are coho salmon from the Central California coast, ESU, which are critically endangered. We are back in the Russian River, which you just heard about from Mariska. So that is right here on this map. It's the largest watershed within the Central California coast. ESU historically supported the largest populations of Central California coast, coho salmon, and now there are very few wild fish left in the river network after sort of decades of habitat change. But there's also a lot of momentum for conservation and restoration, including a large conservation hatchery program and a huge monitoring network. So what you see in those red dots are a bunch of pit tag antenna arrays, and there have been hundreds of thousands of juvenile fish that have been pit tagged over the last 12-ish years. So lots of data, fairly large spatial scale. Good job, Mariska. So I'm going to be talking about intrapopulation variation in movement phenotypes. I don't know if you all care about this, but it's not just in coho salmon. Intrapopulation variation in movement is ubiquitous in nature, from microbes to fish to turtles to elk and insects. And I guess I put that in there just because I think there's a lot we can learn from ecologists across the board, and a lot of our information might be useful to folks who study things like elk. Why do we care about this? There's very good evidence that diversity begets stability. So as with a financial portfolio, diversify, especially in these very trying times. The canonical example in sort of our fishy system would be sockeye salmon in Bristol Bay, Alaska. So this is from Schindler and colleagues looking at distinct populations in those skinny lines being much more variable than the aggregate metapopulation in the thick line there. So you can see that's flatter than the spaghetti lines. And so I think that's fairly well known, and I think the concept of portfolios is pretty intuitive to a lot of us. You spread risk out across space, but what we were wondering is sort of can this operate at much smaller scales? So I was funded for my postdoc to work on Willow Creek, which is this beautiful and very interesting little watershed Mariska covered in part in the last talk. So that's down here. It's the lowermost major tributary to the Russian River. Its mouth is just its confluence with the Russian River is just a couple of kilometers from the mouth of the ocean. It's super interesting. So in the sort of upper portions here, let's say, other side. So in the upper portions here, it looks kind of like what you might think a coho stream should look like. It flows through a second growth redwood forest. There's lots of shade, riparian cover, spawning gravels. There's you can see some structures that have put in to try to kind of regain the classic meander that we're looking for. But then right here, where there's a pit tag antenna, just below that, it kind of flows into this broad alluvial valley where there's been sediment aggregation for decades, creating this very flat and wide valley. The river sort of spreads over that and forms this very dynamic network of channels that kind of braid and reconnect, and they, as the stage rises during the wet season, it creates these kind of like off-channel pond habitats, inundates terrestrial vegetation, and so we think it might be really nice rearing habitat for these fish. So this is a Foodscape session. We think that, as the water rises and it inundates terrestrial vegetation, there's all these things like spiders, and there's tons of terrestrial nutrients that are going to be flowing in. It's much warmer. It flows through an alder forest, not a redwood forest, so it's getting more sun. It's very productive. And then in the lower portion of the tributary flows into this tidal wetland that we also think might be really productive. So for this portion of the talk, I'm going to be talking about that lower portion as non-natal habitat because it doesn't have any spawning habitat, so that might be a little different from what we typically talk because it's still within the same creek. And then up above, we're going to be talking about the natal habitat as the area that contains spawning. Sorry, I'm flying on about 14 cups of coffee right now, so. Okay, so the first thing we wanted to do is just characterize movement patterns within these creeks. So we have a ton of pit-tagged fish, and what I'm going to be talking about is down migration is when fish pass from this natal habitat past this pit-tag antenna right here into what we're calling the non-natal habitat. And then what I'm going to be calling emigration is when they leave the creek into the Russian River estuary. So a couple of, and we're just going to introduce sort of the pit tag system that we're using. You all know what pit tags are, I think. A couple of research questions. So do juvenile coho salmon in Willow Creek exhibit variation in movement phenotypes? And spoiler alert, they do. So the next question that we are interested in is sort of what are the consequences of this for population dynamics? And finally, can we sort of predict this based on things like flow or interspecific competition if it varies through time, which it does? Yeah, so I'm just going to jump right into some results now. Okay, so this is just to remind you sort of what's going on over here. These are our results. So this, on the x-axis here, you have water year day, so days since October 1st. And then you have just histograms or frequency plots of when fish are down migrating, so moving past this antenna here. So you can see there's this really distinct multimodal pattern here that turns out is actually bimodal within years. And so we used a technique that I can talk about if you're interested to classify fish based on this pattern into either one of two sort of modes, constituent distributions. And so these ones here that down migrate in November, December, January are going to be called non-natal because they're down migrating early and spending lots of time in the non-natal habitat. And then these fellows here are the ones that exhibit sort of the canonical natal life history pattern. So next we're interested in sort of, all right, we found this striking bimodal pattern. There's a bunch of non-natal rears, there's a bunch of natal rears. We can see that, so this is just proportion on the x-axis or on the y-axis. For juveniles, the natal fish, which are in yellow, make up about 60% of the population. But they are, the non-natal fish are actually overrepresented among adult returns. And so you can see that they make up over 60% of the adult returns. So this non-natal strategy seems to be pretty effective, and even more so than the natal strategy. And this is over, I don't know if I mentioned, over 9 cohorts over 12 years of data. So quite a few fish. Not very many fish returning as adults, as you can see. So next we're interested in looking at the stability of adult returns and how sort of this variation affected stability. So on the y-axis here we just have the inverse of the coefficient of variation, so higher up is more stable through time. And if you look at just the natal rearing, you can see it's by far the lowest. Non-natal rears are much more stable and sort of confer stability to the population as a whole, and this is a pretty strong effect. Okay, so why is it more stable? We think it might be mediated by variation in immigration timing. So the non-natal rears leave the stream earlier, but much more variable in time than the natal rears. So this might be sort of another level of a portfolio effect where you get more variation, which helps them, it spreads risk out and helps them may help them time their departure to the ocean with sort of favorable conditions, which we all know is very important. And there may be, this is the most Foodscapey part, it may be mediated by differences in growth as well. So this figure is looking like some things that I've seen other people put up today. So they have similar mean growth or median growth, pretty much identical, but much more variable growth, including higher maximum growth in the non-natal habitat. Cool. So next we're looking at predictors of variation. So the number of fish that exhibit this non-natal rearing strategy varies across years, and we were trying to sort of understand why. And so we came up with a couple of hypotheses. One is that higher fish densities increase the proportion of fish that do this. So this is sort of the idea that like there's a limited carrying capacity in the natal habitat. There is some sort of competition that takes place and the losers get kicked downstream. And then the other idea is that higher flow increases the proportion of fish that do this. So that may be that, you know, there's higher flow, there's better habitat downstream. It's easier passage downstream, so passage is kind of limited in this system. So those are the two ideas that we tested, and I'll talk about flow first. So this is flow on the x-axis and the proportion of fish exhibiting non-natal rearing on the y-axis. This is a beta regression curve. I think it's an okay fit, but what you can see very clearly is that in the 2 years where there's super low flow, there are almost no fish that do this non-natal rearing strategy. And then the number of fish released from the hatchery is our proxy for intra-specific competition. You can see there's not really a strong relationship here, so we actually don't really find evidence that there's sort of this competition-mediated driver of non-natal rearing behavior. So some quick conclusions from this section. Juvenile coho exhibit a pretty striking bimodal pattern of variation in down migration timing, even within this sort of small creek. Non-natal rearing increases the frequency and stability of adult returns, so this seems to be a very good thing. And this may be mediated by differences in growth and immigration timing, sort of adding new layers of diversity to this population. But they don't do this in years where there's very little flow. So, you know, all the bad things that drought brings to fish populations may be sort of compounded by this loss of phenotypic variation that we think is really important for these fish. So I'll just put a plug for our recent paper, sort of documenting these if you're interested in it, in Ecology Letters. Check it out, please. And that brings us to part 2. So now we're going to zoom out of Willow Creek to sort of more of a watershed or riverscape scale, and sort of try to understand, you know, Mariska showed that a lot of these fish leave their natal streams altogether. They don't just move to the lower portions, but they leave and they find, many of them try to find somewhere else within this river network to rear. We know that they do this. We don't really know where they go, or why they go to where they go, or when they go to where they go. So that's sort of what we're trying to do, leveraging this huge data set that's been collected over many years to try to draw some robust inferences from this. So we're sort of interested in stream gradient. I'll just note that the 4 streams that Mariska was talking about as sort of her focal streams in the previous presentation are also going to be our focal streams here. So Mariska talked about valley bottom area, but I'm going to be talking about gradient. They're correlated in the stream, so low gradient streams also have a larger valley bottom area. And then also position within the river network. So these highlighted streams are our focal streams here. You can see Willow, which is the one I just talked about, is right near the ocean, and then you sort of get increasing distances away. So the idea there is just that, you know, more fish have to pass Willow than have to pass Mill Creek on their way to the ocean. Maybe that affects non-natal rearing. Okay, so I mentioned that it takes big data sets to talk about these things. The data set that California Sea Grant, Snowwater et al. Have been building for a long time is, you know, over a million, almost 2 million records of almost 400,000 fish. And then when you filter for fish that do non-natal rearing in one of our sort of 4 focal watersheds, we ended up with 31,000 observations of 3,000 fish, so pretty good sample size, but again, that's 0.73% of all fish. So just kind of hammering my point home that these kind of large-scale monitoring efforts are really important for gathering these types of information. Okay, so I'm just going to jump into some results now. So we have sort of two sets of antennas within each of these creeks. The lower antennas are right near the confluence with the main Sam Russian River. The upper antennas sort of separate the spawning habitat, so they're several kilometers up the stream. And then here we have on the x-axis our 4 streams with increasing distance from the estuary. And then I'm going to be talking about sort of a standardized number of non-natal rearers across our entire 12-year data set. So this is standardized, so don't pay attention to the number on the axis too much, but you can see that Willow Creek has, like 5 times as much, as many non-natal rearers as all of the other sites, and that is sort of irrespective of the stream gradient. So Willow Creek is positioned right near the estuary, and it takes in a ton of fish. For the upper antennas, you sort of see a different effect. This seems to be driven by gradient. So, okay, now do pay attention to the axes. You can see that 0.02 is much smaller than 0.5. So most of the fish that enter as non-natal rearers don't go into the upper portion of the tributaries. Most of them are staying kind of in this lower portion. But of the ones that do, they really only go up there in low gradient systems. That makes sense. It's hard to swim up a high gradient stream, but some of them do it, which is cool. Okay, so next we're looking at the phenology of non-natal entry, so the timing of the year at which they enter these streams. The x-axis is sort of this estimated age at non-natal rearing, or days since March 4th of the year that they're born. And we can see that there's sort of these 3 distinct modes. I don't know if you all can see this one. I forget that cyan doesn't show up well on these things, but there's another peak over there. And they correspond roughly to different life stages of fry, par, and then pre-smolts. I think it's really interesting that it's not just sort of a unimodal continuous distribution, but that they get these distinct modes. Okay, and next we're asking if stream gradient affects the phenology of non-natal habitat use, or if position within the river network does. Okay, this is not labeled well, but this is increasing distance away from the estuary here, and these bar plots are the same data as shown here. And then the dashed ones are low gradient streams, and the solid ones are high gradient streams. And what I want you to take away from this is that the low gradient streams have a later date of entry and a later date of departure than our high gradient streams. And there doesn't seem to be much of an effect of position within the river network for this. The other thing I want you to see is that it's again not a unimodal continuous distribution just being shifted over, but that there's 2 modes for each of these. And the reason for that is that it is sort of different life stage compositions that are being attracted to these different sites. So Willow Creek and Green Valley Creek are low gradient streams, have many more of these pre-smolt fish that are coming into them, whereas our high gradient streams attract proportionally more fish at this par life stage. And very few fish that are being tagged are moving in as fry, even relative to the number that were tagged, which is fewer. 1 minute, okay. We also found, so we looked at the time of day of entry. We didn't really have any sort of a priori hypotheses about this, but I thought it was interesting, so I want to show you guys, maybe you have some ideas. So I think that most people think that juvenile fish migrate at night, to avoid predation. So we tested that, and we found that sort of indeed there is a dip. There are fewer fish that are moving during these daytime hours. But what's interesting is that really only seems to be true for Dutchbill and Mill Creek, which are our high gradient streams, and it doesn't really seem to be true for our low gradient streams. So those are QQ plots, and the dip shows that there are fewer than expected under a uniform distribution. We don't really know what's going on with that. We think maybe turbidity has something to do with it. Let me know if you have any ideas. And then just some conclusions from this. Non-natal rearing is most common in streams nearest to the mouth by a lot. Stream gradient affects the timing of entry and location within the stream of non-natal habitat use. And then just to kind of hit home our key takeaways again, juvenile coho salmon are more diverse than is typically appreciated, and even small scale or cryptic diversity can have large effects on population dynamics. We should really be thinking about this for conservation and restoration planning. It seems to be really important. Everyone here has talked about various reasons why it's important. Part of it is we need more big data sets to understand its importance. So just acknowledge a bunch of people who have mentored me or have worked on collecting these data, proof that I've been to my field site, contact information. Yeah, thanks. I'll take questions if we have time.
Hello. Thanks for being here. My name is Amy Fullerton. I'm with NOAA Fisheries. I wish I could be there with you in person too, but as you may have heard, we're not traveling these days, so I hope the conference is going well. So today you heard a lot about foodscapes, what they are, and how we can leverage foodscape thinking to improve conservation for salmon and other riverine fishes. This talk's going to take us a step further and encourage you to think about how we collect and interpret food data with more of a fish-centric focus. So Gabe Rossi and colleagues, many you've heard from today already, or in the audience probably, have a while ago gathered a bunch of smart people at a conference and began to coalesce ideas about what they termed foodscapes. That foundational paper came out in Bioscience last year, and I'll go over its basic tenets in a moment. The second paper, the focus of this talk, was born from discussions with many of the same authors as we wondered how we would implement some of the foodscape concepts given existing data limitations. So we worked together to make sure our paper fit glove in hand with the first to complement its message, but to focus more on the methods aspect. So the original paper, Rossi et al. And Bioscience, had three major hypotheses, and I'm sure you've probably already heard some of this earlier today. The first is that there can arise growth hotspots throughout the river, the riverscape, based on the different kinds of food that's available at different habitats, different times, due to asynchronous events and cycles that determine those availabilities. The second one is that fish are mobile and they can swim to optimize the food that they encounter, right? So they can profit by tracking the peaks in growth potential across these riverscapes. And third is that putting those together, diverse foodscapes can beget diverse life history strategies for mobile consumers like fish that use the spatial complement of habitats and food webs in different ways. So for example, the little image there came from their paper and is illustrating that fish that grew or that were able to access the floodplain in California were able to grow better because they had higher caloric food available to them than the fish that were only able to forage in streams. So this third part is not something that we get into in our paper, but I just wanted to lay that out as a basic overview so that we're all talking from the same place here. So here's an example to help illustrate what this looks like. So food productions in different habitats is not static like we assume in a lot of models. So here we're representing different habitats by headwaters, main stem, and floodplain springbrook. And the temperature can be really different in these different places. So cooler in the headwaters, warmer in the main stem, really cool where there's groundwater influence perhaps. And yet we still assume that food production is the same across habitats in a lot of our models. But we know that this isn't true. We know that the abundance and accessibility and quality of food, these are elements of food, and I'll talk about that again in a minute, that these can differ among different habitats through time. And when you put this together with a fish that can move, it has implications for the growth that's possible in these different habitats. So looking on the right panel there, if a fish were to only stay in the headwaters, it wouldn't grow very much. It's more variable in the main stem, and maybe they grow a lot in the floodplain. But a fish that can move among all those habitats can possibly grow the most because it's able to track what is most productive over time. So before we talk about any methods and taking the next step here, let's talk about definitions. As we see it, there are specific components of food availability, including quality, abundance, and accessibility. Rossi et al. Added physiological growth potential as well, an element we don't cover in depth, but I'll mention it here. So what we mean by food quality are things like energetic density, caloric intake, how easy is it to digest something, nutrient profiles, things like that. And those are represented in our funnel here by different kinds of food. So small terrestrial insects, large terrestrial insects, soft-bodied, hard-bodied aquatic insects, salmon, eggs. These have different amounts of nutrient profiles and quality. And then the next level is abundance. So just total amount of food that's available in a habitat. And that can differ in different habitats. Maybe a riffle has a lot and a pool has a low abundance of a particular type. And then the next level in the funnel here is accessibility. And here we're starting to talk about how a fish can access their food. So can they capture the food? Can they actually physically access it? Are there predators or competitors perhaps that are limiting their ability to get a hold of that food and ingest it? And then the final piece is, what are they going to do to assimilate that food and actually bring that into their body to grow? So just starting with a common framework of definitions. The questions that our paper addressed include: why is food availability challenging to quantify? 2, what are the consequences of uncertainty in food availability estimates? And 3, what methods exist for quantifying food availability? So for the first one, why is it so challenging to quantify? If we think about the way that food information has been collected over the years, it's usually been a focus on stream community composition or just understanding biomass and productivity in the stream. It's not really been collected with the purpose of understanding how food benefits fish. So there can be a potential scale mismatch. It's like looking through holes in a piece of fabric. The way that the data have been collected are very intensive and identifying things down to the species or genus in just a couple of samples once per year, a couple seasons per year, a couple locations, but it's really not giving you a picture of what that foodscape looks like for a fish that can move around. There's also a potential mismatch in the metrics that we used with those approaches. For example, the IBI, the Index of Biotic Integrity, and it may not align well with what aspects of food are important for fish. There's a lot of variability in the foodscapes and what drives them, and these things aren't very well understood. Is it temperature? Is it nutrient availability for the prey themselves? Things like that. And prey quality is really hard to assess. So we just don't have a lot of understanding about that. So, all this means extrapolate at your own peril. It's really difficult to scale up because this relies on so many assumptions that are difficult to validate, such as that food translates to fish growth, survival, and reproductive success. Much of the habitat restoration literature focuses on physical habitat, and we're beginning to understand the importance of water quality elements like temperature and toxics. However, without considering food availability, conservation efforts can be misguided, and management strategies risk failure. The literature is varied about whether food is the dominant feature controlling fish production, and this partly stems from the shape of the bioenergetic response curves, like metabolism, consumption, and growth. For instance, when temperatures are in the range where response curves are steep, like really low temperatures, really high temperatures, temperature may be more important than food limitation. Whereas when temperature is within a tolerable range, like the flat part of the curve, or the top part, food availability can be really important. So conditions when food is likely to matter for populations include when food is limiting, under, for example, oligotrophic systems, or when there's high density dependence. And food is less likely to matter when factors such as low habitat availability or high temperatures or stressful biological interactions are more limiting. Both thermal and prairie resources vary across a range of spatial scales, including reach, watershed, and landscape, which is the left column, and temporal scales, including hourly, seasonal, and decadal, and that's the middle column. Variation in prey resources are available to stream fish, and it's affected by biological interactions like stream flow and habitat, in ways that are really poorly understood. But interactions between food and temperature can create upstream-downstream gradients, which is what's shown in the upper and the right side panels. So misunderstanding which is limiting in a given season or a given year could lead to inappropriate management interventions. So for example, maybe temperature is more limiting in certain situations, certain seasons in the summer, but food helps buffer that in the winter. And having an idea about what kinds of things contribute to those in different situations will really help us to craft management interventions that are going to work better. So what methods exist for quantifying food availability? We present a hierarchy of approaches from coarse and inexpensive to more complex and expensive for characterizing food for fish. So at the really simple, coarse, less expensive end, we have things like eDNA sampling, which gives you more presence-absence than anything at this point, but that's developing. There's bulk sampling, bomb calorimetry, trait-based or energetic-based taxonomy. So these are things like collecting samples, but then instead of identifying them down to a very fine level, in terms of taxonomy, maybe lumping them together functionally by traits or energy or other aspects of food that could be important for fish. And then there's the Riverscape approach, which is really taking less intensive samples more frequently or in more places over space. There's diet sampling, which could be argued as a really great idea for understanding food for fish, but it also has caveats, and these include things like some of the invertebrates are soft-bodied and they're metabolized or digested, I suppose, really quickly. And it also doesn't necessarily tell you that what a fish is consuming is the best condition for the fish. It may just simply be eating what's available. It may not be having much choice in what it's consuming. Then at the next level is the traditional site-based prey sampling we spoke of already, more for the community analyses. Some newer approaches and molecular techniques. Stable isotopes been around for a while and can continue to tell us a lot, but there's been some new research starting to understand fatty acids and those profiles and what that means for fish nutrition. And then at the really maybe more complex end of the scale, but really is one that's necessary, I think, at this stage while we're still data limited, is trying to think about how do we predict proxies or heuristics to characterize what a foodscape might look like based on first principles and things that we know might be driving those. And I have an example of that I'll talk about here in a minute. And then combining approaches might be the best way to go here. So diet sampling definitely tells you what the fish are eating, but it doesn't tell you what's available in the environment. So combining it with some kind of environmental sampling might be more productive in understanding the foodscape and what's available to the fish. And recognizing that there's caveats with each. So I'd like to outline a couple of future directions here that my co-authors and I outlined in the paper just to leave you with. And the first is that we maybe need to start thinking about developing some sampling methods that are really targeting food for fish, and that can be used to accurately characterize foodscapes, so that they can be compared across different study designs or different scales. We need to think about what metrics of available prey really mean for fish, and how do we convert samples of abundance, for example, into metrics of available prey. Like, what is something that fish can actually consume and use to grow? And just thinking about that, there's all like that information funnel I talked about earlier. There's a lot of different ways of terms, I guess, out there floating around about how to think about food for fish, and it's really worth thinking about the metrics in particular. Understanding how other factors affect prey availability can be really important. So what are those drivers, and in what situations are they going to be more important than others? And starting to produce some predictive models of food availability for use in conservation and management, because we just don't have a lot of empirical data right now. So we need to, use what we have and our understanding and our ecology to insert into conservation and management, because we can't ignore food. It's a really important piece of food conservation in addition to just habitat. So another thing that we can do more of, and there's already a lot of this, is to conduct more integrative modeling, like bioenergetics, individual-based model, simulation type things, to understand the importance of the variance in prey availability to individual fish, and how they grow, how they survive, and how that translates up to population persistence. It's a really useful tool, again, in the absence of having a lot of really good data. And another opportunity is there's a lot of restoration projects happening on the ground. And I know that this is a whole topic, restoration effectiveness monitoring, but I think what I mean by that is more habitat-based, and it would behoove us to think about how these management actions are actually affecting the prey available to fish and how they're restructuring food webs. So there's an opportunity. And then I mentioned this earlier, but I just want to hit on this again, this concept that there may be cases where understanding food availability is really important and we need to do better at it. And there may also be cases where it just doesn't matter. Like it doesn't matter because something else is more limiting. And so this is kind of maybe a first step for any assessment, is just understanding when and where we need to really be thinking about food. So before I end, I want to give you a note of hope here that we're making some progress on some of these things by highlighting a project. So this project is actively ongoing. This is work by Peter Kifney, Elsa Toski, and Toshio Matsuoka, and they have been working with a lot of other researchers in the field to assemble a large data set of drift invertebrates so that they can support foodscape modeling. So they're trying to quantify the spatial and temporal variability in stream invertebrate drift, which is a major source of energy for drift feeding fishes. And they've collected all these data. I'm going to show you this on a map here in the next slide. And they're working on 3 objectives. So the first is a paper that's already in review, just quantifying the data, basically describing the data, the metadata, the spatial and temporal variation in food availability, food concentrations. The second is a paper that's going to examine the role of both local and landscape drivers on stream drift. So local could be things like temperature, benthic invertebrate abundance, etc. And landscape drivers might be things like fire or geology. And then the third product or phase will be trying to figure out how to incorporate this information, this food availability information, into a salmon life cycle model. It's called the Habitat Assessment Restoration Potential Model. And to increase the realism of that, just really by bringing the food in and thinking about what physical and chemical drivers that exist, but also what food drivers exist and how that can affect our ability to recover these species. Okay, so the map here, it shows the distribution of the sample reaches, which range almost 40 degrees of latitude and 10 degrees of longitude. Most sites are in the marine west coast forest, in the cold desert ecoregions, and in the western cordillera. And I would say if you have information about other data sets that you might be willing to share with Peter Kiffney, please do reach out. Okay, that's all I have. I think you'll agree that assessing food availability is not simple, but that there are steps we can take to begin to think about how we assess foodscapes by focusing on the fish. So sorry I can't be there to answer questions in person, but please reach out by email if you have any thoughts. We'd love to hear them, or to the first author of the paper, Valerie Ouellette, or any co-author. Several of them are probably in the room. Thank you very much, and enjoy the rest of the conference. And thanks, organizers.
So the title of my project is River Rest Stops, the Effects of Floodplain Food Subsidies on Chinook Salmon Outmigration Transit Times. It's a bit of a mouthful. And currently my research is funded by Cutthroat and the Bureau of Reclamation. So a little bit of background that you've all heard all day, but I got to go through it anyways. Historic records show that California salmon has been in decline since the 1920s. And currently in the Central Valley, the numbers are less than 75% of their previous numbers. It's not looking good. And specifically late fall salmon run are listed as a species of concern. Spring run is listed as threatened, and winter run salmon are currently listed as endangered, a little bit of more cause of the decline. So one of the biggest causes of decline in salmon is channelization of the Sacramento River. Up there we got a photo of the Sacramento River, and that's the main channel, and it's confined to 2 levees. Fortunately, we can't take down the levees or we'll flood everyone in Sacramento, but essentially the only way, in the past, historically, salmon were able to access the area outside, basically valuable floodplain habitat, and nowadays they can only really get to the floodplains like the Sacramento bypass when water is high enough to go over the weirs. So those fish no longer have access to valuable rearing habitat and basically all the food habitat that they had historically. No access to the floodplain equals no food. I guess you heard a little bit about the Fish Food Project, but one of the ways that we're trying to reconnect fish with those resources is through Trout's Fish Food on Floodplain project. So in 2017, California Trout launched their Fish Food Project, working with farmers and landowners to flood their fields when they're not being in use. So rice fields, when it's not in season, they flood those fields, and those are prime conditions to grow high densities of zooplankton, specifically like large body cladocerans. That, like in this photo right here, that's a Daphnia magna. It's huge, and fish love to eat them. So the high residence time in those fields is kind of what allows food to grow so rapidly, and essentially when they're not in use, the farmers are flooding those fields in different cycles, and when the density of bugs is high enough, that water is then exported into the river with these large agricultural pumps right here. You kind of see, that's all floodplain water being dumped into the Sacramento River at our main pump site. So yeah, so the project has shown great potential to basically bolster some of those depleted food populations and help out struggling fish in the river. Specifically for my research, the water pump, the export site it's called the Rough and Ready Water Pump, and it is north of Knights Landing, California. Right here we can see on the right hand side is the main channel, Sacramento River. In the middle is the pump house. That kind of white building is kind of a little bit difficult to see, but that white building is the pump house, and it's capable of actually pumping a good amount of water. There's about 6 underwater pumps that can throw out a lot of CFS, and then there's one above water that's basically on a float switch. And then right here, the canal is all the floodplain water. That's coming from all the rice fields that are being flooded. Yeah, I wish I had a picture that showed all the rice fields, but there's lots of acre foot of flooded fields in this. And so yeah, so that is all floodplain water, pump house, dumped into the Sacramento River. So currently, a lot of the support for the Fish Food Program is through caged fish data. So right here we have a picture of some of the trout cages, and inside of each cage there's 5 salmon, and weekly measurements are taken, so they're weighed and measured every week. And at the end of the water export season, growth rates are calculated. However, there's not too much data on free swimming fish around the area, so that's kind of where I wanted to come in and research more fine scale movement data for fish who can actually move around and have a little bit of free will, and they're not just in cages. So the big picture, what I hope is, this is really a big picture, if fish are utilizing these food resources, I think that could better inform like salmon management practices. I think if the data shows it, I believe releasing salmon during periods of like high productivity in the river is basically setting up the salmon for success. If we're letting the salmon go when there's a lot of food in the water, they have the highest chances of making it to the ocean, which is what I hope. To kind of research this, we asked 2 main questions. Why did do floodplain food subsidies have an effect on salmon outmigration time? So is all that food that we're pumping in the river actually changing their times? Are they? And that's kind of the approximate that I used to show if the fish were actually eating the resources. Basically, the time it took to go through the array. And more specifically, will outmigrating salmon spend longer time in subsidized portions of the river compared to other portions? My research was kind of a 3-parter. First was an acoustic telemetry array. Next was weekly zooplankton sampling, and finally weekly fish growth monitoring. So the array was to tell the transit times of fish through our array. The zooplankton sampling was to see if we can pick up the signal of that floodplain in the river. And then lastly, the fish growth monitoring, more fish in cages, just to see the growth rate of the fish along the river when it's being subsidized with floodplain water. So here's the stretch of river that I used for my array. On top is the control reach. So in the middle right here is where it says export site. That's where the rough and ready water pump is. So this is all upstream of it. So it's not going to be hit with the subsidized, with the fish food subsidy. But all this down here, the subsidized reach, will be hit with the floodplain water. So we had pairs of receivers at the upstream site. One, we had a pair of receivers at directly at the water pump. A pair of receivers down there. So the receivers we used technologic receivers. We put out more than we recovered. It's kind of, receivers are kind of funky like that. When it's really wet, it was a really wet year, and then trees sometimes they go over the steel cable that you use to put them out, and they get really difficult to recover. But so we recovered 5 of them along the array, and additionally I downloaded some receiver data from the RDAP database for some receivers that were below China Bend in the upper portion. The total river kilometer of the array was 13.18 river kilometers long, and we deployed them in January of 2023. For fish, we didn't actually have to tag our own fish. We kind of piggybacked off of NOAA Fisheries' seasonal survival hatchery experiment, which was pretty nice, not having to tag our own fish. And those fish were released in Red Bluff about like every 2 weeks, I believe, during our water export period. For our zooplankton sampling, weekly samples were taken with zooplankton throw nets. This picture right here is one of the technicians with a 30-centimeter throw net. And once the zooplankton samples are collected from the river, they're then put into bags like this, dyed with ethanol. And right here, it's kind of hard to see, but if you've taken zooplankton samples, this is a pretty actually dense sample. It kind of looks like dirt, but that's all big bugs down there. So that's actually a pretty good sample, yeah. So weekly samples were taken, then taken to the lab, where they were painstakingly counted to calculate the zooplankton densities along the river sites. Next would be the weekly fish growth. This is the cage fish. This is a nice picture of a fish being measured. And then on the picture to the right is the crew weighing the fish. And so these, like I said, these were used to calculate the growth rates at the end of the water export season. So into some of the fun stuff. So some of the 2023 preliminary results. I also had 24, but 24 we had kind of a malfunction with our receivers, and we only picked up like 20 fish in our array, so it wasn't very good. So this is the 2023 data. So as far as zooplankton abundance in the river, that large red spike right there is directly outside of the water pump. So that's going to be the largest densities of zooplankton in the river. And then the green lines are all within the subsidy reach, so they're all different sites along the subsidized portion of the river. And then the orange lines on the bottom are going to be the density of zooplankton in the control reach, so north of the water pump. And you can see right here that at the water pump it was much higher than the control reach, and then also higher than other ones downstream. As far as growth rates, this picture might have been shown today, but at the floodplain outlet, so directly outside that pump, the growth rate of fish was 7 times greater than the upstream sites, than the fish in the control sites. And even as far as 6 miles downstream, the growth rates of these caged fish were still higher than the control site, kind of showing that even 6 miles down, they're still seeing that subsidy, and these fish are still getting all those benefits. What we're not able to tell with those cage fish is if we can't tell what the free swimming fish are doing. So in order to tell if the free swimming fish are using the subsidy, we used fish ground speeds to the array. So over 200 different tagged fish were picked up in our array, and I have some cool examples to show y'all of a fish transit plot. So this is a waterfall plot of fish moving through our array. You can see this orange line showing that denoting the river flow. And then on top of this red dashed line is our unsubsidized zone. So you can think of that as upstream, and then it just goes downstream as the arrow goes down. The bottom part is our subsidy zone, and then that red star just shows where the pump is. So you can see a fish like this was picked up at the top of our array March 10th. And then it kind of made its way. This would be considered a pretty fast moving fish. Well, at least I consider it that from my data. Because it only spent around 8 hours within the array, so it was first picked up on March 10th, and then 12 hours later it was out of the array already. So it traveled the whole 13 river kilometers within 8 hours. It didn't really stick around for any reason. We also found a good amount of fish with about a medium transit speed through our array. So this fish was picked up also on March 10th, a busy day. And then this one actually spent a total of 1 day and 6 hours within our stretch of river, which is really cool. So it was picked up March 10th, and then it got picked up directly at the pump about 12 hours later, and then it spent about a day in the subsidized portions of the river before it finally left our array. And finally, what I think is a really cool fish is this one right here. So this one was picked up earlier in the season. This fish was picked up on February 13th, and it decided to, first picked up in our control zone on February 13th, and then it was picked up like a couple hours later in our right at the pump, and then it lasted about a month in the subsidized portion of the river directly around the pump before it decided to leave our array. This fish was definitely surprising, so I did a lot of cross checking. I looked at the entire fish movement data to see if this was a real fish and make sure it didn't just get eaten by a striper or something. That could happen. But yeah, it was real. It was a real fish. The timelines all lined up for the entire movement history of this fish. So it's really cool to see just the different strategies that these fish could possibly be using as they're traveling through this array. Finally, I calculated the transit times of the fish through the array, and a little bit surprising to me, the average fish speed above the pump, so in the unsubsidized portion, was a little bit slower than in the subsidized portion. So above the pump, on average, they swam at a speed of 2.36 kilometers per hour, and then the average speed below the pump, they swam at a speed of 2.63 kilometers per hour. So then you can see this block spot right here. This one represents the fish speeds above the water pump, and then the orange one represents the fish speeds that were below the rough and ready water pump. So I looked a little more in depth into this, and I decided to check the fish speed only in the subsidized portion of the river, and I used the variable when the pumps were actively exporting water versus when they weren't exporting water, which is when we start to see kind of switch. When the pumps were actively exporting water and the in our reach of the river, the average fish travel speed through our array was 2.52 kilometers per hour, versus when the pumps were off and not exporting water, they traveled a little bit faster at 2.86. So there's a little bit of an effect when the pumps were on. And even more specifically, I looked at the fish speed through that subsidized portion of the river during the largest zooplankton spike. So out in the corner is the graph I showed before with that large red spike showing a large subsidy of zooplankton in the water. And so when I just calculated the fish speeds of fish around that time versus periods when there wasn't as much subsidy in the water, there was a much larger difference in transit time. So on average, during a large subsidy spike, the fish traveled at 2.18 kilometers per hour. And when there wasn't as much food in the river, they seemed to travel, during the unsubsidized portions of the time, they seemed to travel at faster at 2.84 kilometers per hour, which was pretty cool to see. So it's very preliminary, all my analysis and stuff, but it does seem that fish do seem to be utilizing some of these food resources that we're providing. It seems that fish have lower ground speeds during periods of the largest food subsidy in the river. There's some really cool extreme examples of fish who spent a long, long time around these pumps, which is really interesting to see. But there's still a lot more I would like to look into. I definitely need to include river flow effects into these transit times. I have all that data, I just haven't had the chance to look and present it yet. I'd also like to look into the fish size at release to see if that had anything to do with the fish's strategy of its outmigration. I have a couple hypotheses that maybe if the fish was larger at the time of tagging, that maybe it didn't really need to spend as much time around these food pumps. Maybe it was ready for the outmigration and ready to make it to the ocean already. But I'd have to look at the fish tagging metadata and kind of do my own analysis of that as well. And lastly, I want to compare the outmigration success of fish that were around during times of large subsidy in this reach versus maybe fish that passed through and they might have missed the signal. And I want to compare the success of those fish if they made it to the ocean or not. I think that'd be really interesting to see. Yeah, so that's all I have so far. Thank you to everyone who's helped me on this project at UC Davis.
And thanks everyone for sticking it out to the very end. For my presentation today, I'm going to talk about two projects that are focused on salmonid ecology in streams and food availability in streams. So in streams, salmonids are thought to gain the most of their energy foraging on invertebrate drift, which is the downstream flux of aquatic invertebrates, aquatic and terrestrial invertebrates in the water column and then also on the water surface. And as a prey source, it has a couple of very interesting properties. One, it's continually renewing or continually generated and delivered into the foraging area. And it does not accumulate in the foraging area. So because of this, salmonids have to defend space to maintain access to food availability, to high levels of food availability. And so when they're foraging, salmonids behave as central place foragers. They're visual predators, so they forage primarily in the daytime. And so they'll hold position facing into the current and then move and selectively intercept prey items as they're transported downstream into their foraging area. So the predation cycle for a drift feeder or a salmonid can be divided into processes of prey search and then assessment, moving to intercept the prey item, handling the prey item, and then returning to search again. And it's thought that salmonids primarily forage on one individual per foraging bout, but there's some recent data that shows that maybe there's some variation in that. And so despite the importance of food for salmonids influencing their abundance and productivity, food availability has received far less attention than other physical habitat features. But studies that have looked at food have found that it's very variable in time and space. So this figure shows invertebrate drift densities on the y-axis And the study was conducted in the John Day River across 5 streams. The bars that have similar letters are from the same streams, or that have the same letters are from the same streams. You can see that within streams here, and at the end, invertebrate drift abundance is quite variable. And then between streams, it's also very variable. And it's also extremely variable seasonally. So this figure shows invertebrate drift density on the y-axis across years. This was from a stream in New Mexico. It shows drift density on the first y-axis and drift energy density on the second y-axis. And so looking at this figure, we can see that drift density and energy track pretty similarly across the year. And we can see that there's huge spikes in prey availability. And then also there's big lulls in prey availability. So what this means is that salmonids have to capitalize on these during these times of high prey density to maintain energetic reserves when there's very limited prey available. And I just want to point out just how variable drift is. The 2 axes here, so the figure on the left shows drift densities in number per 100 meters, and then the one on the right is just per 100 cubic meters, and the one on the right is just cubic meters. So drift densities span a massive range. So during times of low or high prey availability, salmonids have to capitalize, as I mentioned before, but the rate at which they can do that is ultimately limited by the rate they can complete that predation cycle. So studies have looked at how fast salmonids can complete this predation cycle using Holling's disk equation, the functional response model. This particular study looked at grayling feeding on Daphnia in laboratory stream environments. And so the y-axis is foraging rate, and then prey density in number per liter on the x-axis. And you can see that as prey goes up, so does foraging rate. But the line begins to bend over, and so the rate of increase begins to decline until the fish is foraging as fast as it possibly can, right? It's completing that predation cycle as fast as it possibly can. Again, so the units here I'm going to point out just because it's relevant to the data I'm going to present at the end. Here, this is number per liter, so it's 1,000 liters per meter square, cubic meter. So the highest densities these fish were exposed to is 2,000 individuals per cubic meter of water. And also their foraging foraging rate for the maximum here was almost 1 foraging bout per second, which seemed really high. So I was curious if these prey densities are representative of those we find in natural streams, and if these foraging rates are similar to what we would find in natural streams. Specifically, I wanted to know if salmonids display a functional response to increasing prey availability in natural streams, and if so, what are their maximum foraging rates? And at what drift densities does that line bend over? At what drift density does increasing food no longer benefit an individual fish because it's feeding as fast as it can? So I conducted a study in 5 streams in southeastern Idaho where I used underwater videography. I filmed foraging fish that I located either by snorkeling or bankside observations. I filmed them for about an hour, and then immediately following filming, I collected drift samples right at the foraging location to estimate how much food they've been exposed to. I watched the videos, I measured the I looked at the location they were foraging from, because while they feed primarily on drift, they can feed from the benthos as well. Salmonids can feed from the benthos as well, also from the surface. So I enumerated the number of foraging attempts at each of these categories, and then calculated the foraging rates for those categories. Back in the lab, I sorted the drift samples, and then I measured and enumerated the invertebrates. And the reason I measured them for length and width, is because I wanted to know what was the energetic content of each of those individuals, so I could look at the energetic flux going downstream. So I used this equation to transform prey sizes into joules. And so for this study, I had 15 cutthroat trout. The video durations were from just under 3 minutes to over 36 minutes. I had a wide range of fish body sizes, from young of the year fish to larger cutthroat trout. And this is what's really important, is that I found streams that hosted large variations in drift density. Okay? And then I also saw large variations in fish capture rates from 0.3 captures per minute to over 6 captures per minute. And when foraging, I found that salmonids forage primarily from the water column, with less foraging attempts directed at the surface, less foraging attempts directed at the benthos. Now, because I was primarily interested in drift foraging, and drift foraging, as I mentioned on the first slide, can combine surface foraging and water column foraging. I combined these two. I just added the number of the foraging rates together and then modeled it over prey abundance. And what I found is that cutthroat trout did display a functional response to increasing prey availability. So the top panel here shows foraging rate and captures per minute, and then down the bottom panel shows energy intake rate. And they're both across drift density. You can see that both of them display a functional response, with the model predicting, the top model predicting just over 5 captures per minute, and then that line bending over or being maximized at around 15 invertebrates per cubic meter of water. And for the energy intake model, I found that energy intake rate was about 66 joules per minute, and energy intake was maximized around 24 invertebrates per cubic meter of water. Now these models gave me different answers, and I was curious, 60 individuals per cubic meter of water, is that a lot? And I wanted to know if the 5 forages per minute, is that a lot? And so I went to the literature and I looked and tried to figure out really what's the magnitude of invertebrate drift reported in past published studies. And I found that there wasn't a lot of information on this. So next I asked the question, so what's the magnitude of invertebrate drift in natural streams? And then what's the range and extent of foraging rates for fish foraging in natural streams? And so I wanted to see if there were factors like temperature, precipitation, geographical data that could be used to predict drift abundance across a wide spatial scale. So I conducted a literature survey, looking through past studies that reported drift density that was reported in units of number per cubic meter, just so I could compare, so standardize the units. They had to be collected in daytime. They had to be from natural streams where food availability wasn't manipulated, so no experiments. And after I collected those, I collected some covariates, sometimes directly from the papers, sometimes GIS variables. So the covariates included in the data I'm going to present are latitude, longitude, elevation, 3 temperature and 3 precipitation variables that I downloaded from the WorldClim data set, and also net mesh size, which is it's a critical variable when capturing invertebrates. The size of the spaces in the nets will determine the minimum body size that you can capture. So I used model selection methods using a mixed effects multiple regression model to evaluate which of these predictors is best predicting invertebrate drift. And now I had multiple observations within streams that I thought might not be independent, so I included stream as a random factor using a random intercept model. So to get at the second part, I wanted to know what drift foraging rates had been reported in past studies. So I did a second literature survey and looked for daytime drift foraging rates in natural streams. Again, no experiments. In similar units, so I was looking for captures per minute or captures per second. And what I was really interested in is papers that reported both of these, because I wanted to use those data to see if I could refine that functional response model that I had generated in the previous study. And so for the invertebrate drift literature survey, I found 70 studies, almost 350 drift observations across 142 streams from 23 countries and within 6 continents. Unsurprisingly, the vast majority of observations were in North America, also a lot of observations in New Zealand. So these are two locations where there's a lot of drift foraging salmonids and then galaxids in New Zealand. And sort of unsurprisingly, I found that invertebrate drift reported in past studies spanned a wide range. Okay, but the vast majority, almost 40%, reported less than one individual per cubic meter of water, where the maximum was almost 160 individuals per cubic meter of water. So relative to the maximum, invertebrate drift tends to be quite low, or was quite low in this data set. And from the previous study, I found that those cutthroat that I was watching forage, they were exposed to high levels of drift. So this figure's 10 panels. I know it's overwhelming. I'm just going to step through it. So these are all the covariates from the model selection procedure. I found latitude and longitude displayed curvilinear relationships with invertebrate drift. And they were best fit using polynomial equations. I found elevation, temperature annual range, and mean diurnal range were positively correlated with invertebrate drift. And so temperature annual range is essentially a measure of seasonality. It's variation across the year, variation in temperature across the year. And mean diurnal range is a measure of variation in daily temperature. I found that annual mean temperature, annual precipitation, precipitation during the wettest quarter, precipitation during the driest quarter, and net mesh size were all negatively correlated with invertebrate drift. So in areas that are really warm and really rainy, they had low drift density. And so looking at the latitude figure here, areas around the equator had minimum drift density as well. So the data kind of makes sense in that way, that mid-latitude regions that have more seasonality were predicted to have higher drift densities. And so the global model, including all of these variables, accounted for about 35% of the variation in invertebrate drift that I observed, but the fixed and random effects, so the random effects was stream, accounted for 84%. So this indicates that samples from within streams were far more similar to one another than samples between streams. So it was correct to try to control for that non-independence from within stream replicates. Using model selection, I found that the variables that were the best predictors of invertebrate drift density were latitude, longitude, elevation, and precipitation during the driest quarter, and then unsurprisingly as well, the size of the mesh used to sample invertebrate drift. However, as you can imagine, all these variables are super correlated, right? Precipitation, temperature, latitude, longitude, those things are all very correlated. So I had 7 models within 2 AIC from the top model. And I did some model averaging and stuff. That data I'm not going to present today because the talk went a little long, but I'm happy to discuss later. For the drift foraging literature survey, I found 30 studies that reported over 380 observations of drift foraging rates across 19 species, 14 of which were salmonids, and foraging rates paralleled drift abundance as well, where there was a wide range, but the vast majority of observations were low relative to the maximum. And so the cutthroat trout that I found foraging were foraging at a moderately high rate, but not the maximum rate that had been reported, not near the maximum rate, about half the maximum rate that had been reported in past studies, which was 12. I don't know if you can see that over the podium. So I found 5 papers that reported both invertebrate drift abundance and drift foraging rates that I then incorporated into the previous model I had created. So here we have foraging rate on the y-axis and drift density on the x-axis. The open triangles are observations from the literature survey. The closed circles are data from the Cutthroat study I presented before. The solid line is from the Cutthroat study. The fine dashed line is all the data together. The wide dashed line is just the data from the literature survey. And so while incorporating data from the literature didn't necessarily improve the model, all three models showed this functional response to increasing food availability, whereby initially foraging rates increase, but then the process of completing that predation cycle limits energy intake rates for drift foragers. So I found most reported drift densities were low relative to the maximum, and food and space are thought to be primary limits on salmonid populations. And these data suggest that food availability is likely limiting in many populations of salmonids and other drift foragers. Foraging rates paralleled those of drift densities, where there was a wide range, but the vast majority were low relative to the highest observed values. Latitude, elevation, net mesh size, precipitation during the driest quarter were the top predictors across my covariates, and all these data sort of agreed that mid-latitude regions where many drift feeders are commonly found may have higher drift abundance relative to areas around the equator. But there was far more data from these mid-latitude regions than there was from the tropics. And so for future directions, size is a huge component of the energy budget for salmonids. They're both size selective and gape limited, so they can only eat what they can fit in their mouth. And there's a wide array of prey sizes. So this peltopurlid on the left is less than 1 millimeter in length, and then this odonate is almost 2 centimeters in length. So you can imagine they have vastly different energetic contents. Also, seasonality is critical, right? I showed that in a slide in the intro, and I wasn't able to incorporate seasonality in this study because the vast majority of drift samples were from either the spring or the summer or like early fall. There was very few in the winter, so the data was skewed that way. So I think that expanding sampling into winter months could help get a better picture of food availability outside of these primary growing seasons. Also, I think that we could look at the energetic contributions of alternative foraging strategies, whether that's drift or benthic foraging or search foraging, and then looking at thresholds when salmonids switch from one foraging method to another. I'd like to thank all these people and institutions, and I'd be happy to answer any of your questions.
Great work. That was an awesome day. I loved every single one of these talks. I learned a ton today. My email's, if you don't get the underwear model, you get the real Rossi. My email is on the internet. Everyone, please connect with me. Talk to me anytime. I'd love to continue the conversations we had, and then I'll be at the banquet tonight, so I'd love to catch up with any and all of you.
Thanks, Gabe.
Thank you. Thanks, everybody. You guys rock.